am Biswanath Tripathy, and today we are going to revise the hematology part, mainly the blood and the blood cells, about hematopoiesis and about RBCs, WBCs, as well as platelets, and some of the clinical correlations too. So blood, let's start with blood. So blood is a fluid connective tissue. It is mesodermal in origin. It links organs, so it is called as connective tissue. It has cells, fibers, and matrix. So blood mainly used in fluid transport or nutrient transport. Okay, and disease protection too, as WBCs have a role in immunity and waste removal. So we can write many of the roles. Color is red. So differentiating color is that the arterial blood is mainly scarlet red in color, and the venous blood is purple red. So there exists a difference in color. And volume, about volume in adults, it's about 5 liters and constitutes 8% of body weight. So you can get this data from Canon. And about the pH, it is 7.4, we know, slightly alkaline. The specific gravity, as you remember, we can call it refractive index. It is nearly 1, not exactly. About the viscosity, it is 5 times more than water. So the different proteins and the constituents of blood contribute to the viscosity. We can divide blood into two categories. One is the plasma, which is called the fluid matrix or the tissue fluid. So it is of straw colored. And other is formed elements. So in formed elements we have RBCs, WBC, and platelets. In plasma we have organic substances as well as inorganic substances. Like organic substances that constitutes mainly of antibodies, hormones, enzymes, fats, amino acids, and some plasma proteins. Plasma proteins we know: albumin, globulin, fibrinogen. And about enzymes, we have the famous enzyme carbonic anhydrase, lipase, esterases, etc. Non-protein nitrogenous substances are too in plasma. I'm saying non-protein nitrogenous substances. These are also present in plasma, and the examples are mainly the excretory waste products like ammonia, creatine, creatinine. So ammonia, creatinine, xanthin, urea, uric acid. These are also some of the components of plasma. Okay. So now we have a term in hematology which is called as hematocrit value, or you can say it PCV. So it is majorly the percentage volume of RBCs in blood. About the functions of the blood, we can write it has some nutritive functions like distribution of nutrients and digestion, transport of respiratory gases, oxygen, carbon dioxide, and etc. Waste, transport of hormones, enzymes, water balance regulation. Water is also a buffer, so it has buffering action. Acid-base balance mainly, pH regulation you can say. Body temperature regulation, because it increases the specific heat. Okay. And defensive function, like WBCs we have it. Okay. Now let's come to the main topic, plasma proteins. Plasma proteins. We have three plasma proteins, which is called as serum albumin, serum globulin, and fibrinogen. Albumin has the lowest molecular weight, and globulin is mainly the globular protein, and both albumin and globulin are present in serum, and fibrinogen has the highest molecular weight. We have a ratio called as albumin globulin ratio, which is normally 2:1 in normal conditions. About the separation of plasma proteins, like we have a mixture of plasma proteins and we have to separate it. So we have for separation of plasma proteins, we have many of the methods. Like first we have precipitation method. Mainly the globulin precipitates with 22% sodium sulfate solution, so we can write it like globulin PPT with 22% sodium sulfate solution. And we have salting out method too, like you must have read in precipitation of proteins the reactions mainly, and salting out is a method, in which we can separate globulin into euglobulin and pseudoglobulin. We can separate globulin into euglobulin and pseudoglobulin. And nowadays electrophoretic method is also used, which is done by Tiselius apparatus. And dependent on the solubility, we have another method which we call as Cohen's fractional precipitation method. Okay. Centrifugation is also a method, but nowadays ultracentrifugation is also used. Ultracentrifugation. And we can use chromatography, so gel filtration, chromatography. Mainly it has a tube of beads and the protein molecules are passed through the tube, so it is a type of column chromatography in fact. And about the electrophoretic method, we have another method which is called as immunoelectrophoretic method. Okay, so these are not much important, but you must know it. About the properties of plasma proteins, about the molecular weight, fibrinogen has the maximum molecular weight, and serum albumin has the minimum or the lowest. So the order of molecular weight you can write it. About the osmotic pressure, or it is also known as osmotic pressure, also called as oncotic pressure. Although it is contributed by all plasma proteins, but mainly by albumin. Plasma proteins act as buffers, so buffer action is also shown by plasma proteins. Okay, So about the origin of plasma proteins in embryo, these are synthesized by mesenchymal cells. Okay, and in adults, these are synthesized by reticuloendothelial cells of the liver. Okay, but also synthesized by spleen and bone marrow, etc. So, except the gamma globulin, you must know this exceptional case. Gamma globulin is synthesized by B lymphocytes. Okay. Function of plasma proteins, all the functions like transport mechanism and maintain the osmotic pressure, acid-base balance. They maintain viscosity because albumin has the most viscous, so it is maintains viscosity. It is maximum viscous, so okay, obviously. And coagulation of blood through blood coagulation is also done by the plasma protein, so we can write it in the function. And Rolex formation is done by fibrinogen and globulin, so you can write fibrinogen and globulin. This do Rolex formation too. Rolex is actually a pile of RBCs which are stacked like you may stack coins one over another. So RBCs are stacked like that. So in case of Rolex formation, the blood becomes looks pale. Okay. And some substances are there. These are important for PG point of view, which are called as tryptophan substances. These are produced by WBCs, and these are used for nourishment of tissue cells in culture. Okay. So we have the culture. So in culture we can nourish the tissue cells by production of tryptophan substances. These are produced by WBCs, and in fact plasma proteins help in it. Okay. We have a hypothesis now called as the Starling's hypothesis. Starling's hypothesis. Like we have a capillary membrane. We have a capillary membrane, and the net filtration rate through the capillary membrane is directly proportional to the hydrostatic pressure minus oncotic pressure. Must have remembered that oncotic pressure is also called as osmotic pressure, as we discussed earlier. Now we have an experiment called as the plasmapheresis, or we can call it the Whipple's experiment. In this experiment, it was first shown in dogs. Okay, so blood was removed and RBC was separated from plasma. After separation of RBCs, these are washed in saline. Okay, and after washing saline, we add is solution called as LOX solution. LOX solution was added to the RBCs which are washed in saline and returned to the dog. So actually now the blood doesn't contain the plasma proteins, so the dog comes in a state of shock. Okay, state of shock is caused mainly due to the lack of proteins, and if the dog is fed with protein, the plasma proteins can be restored within 6-7 days. And if fed with proteins and liver is also extracted, okay, the shock persists because liver is the main site of plasma proteins. Okay, now we have a thing called therapeutic plasma exchange. About the therapeutic plasma exchange, this is mainly used to treat autoimmune diseases. Okay, used to treat autoimmune diseases, in which from vein blood is removed and we send it to a centrifuge. Then the blood cells are washed in saline. Plus albumin is added and returned to the bloodstream. Okay. So you must have heard about the plasma exchange in case of COVID-19 was persistent there. Plasma exchange was used. So the principle is mainly the antibody removal. Okay, we can discuss it later, obviously. So in plasmapheresis, we discussed plasmapheresis above. In plasmapheresis, we can treat a variety of syndromes. Okay, like we can treat thrombocytopenic purpura or Guillain-Barré syndrome, or Lambert-Eaton myasthenic dystrophy, you can say, Good-Pasteur syndrome. So these are some diseases which can be treated by plasmapheresis. So let's come to the main topic. We can discuss RBC first. Red blood cells, corpuscles. Okay. In birth, actually, the RBC number is more, like 6 to 7 million per cubic mm in birth. And as we progress through age, in males, it gets retained at 5 million per mm cube. And in females, it even less, 4.5 to 5.5, we can keep a range. RBCs are non-nucleated, they have no organelles, they are biconcave. So the advantage of biconcave sieve is that the diffusion of O2 is equal and the tension is less. It can squeeze out easily through the pores and this is called as dipedesis. So due to dipedesis advantage, the RBC size is like that, shape is like that, biconcave. And about the dimensions of the RBCs, you can get it from any standard book or writing it. It is about 7.2 micrometer. This is 2.2 and this is 1. Volume is about 85 to 90 micrometer cube and surface area is 120 square units. So cytoskeleton, about the cytoskeleton of RBCs, it is mainly found by ankyrin, spectrin plus ankyrin. In absence of spectrin, some books write we have hereditary spherocytosis and some say we have leptocytosis. So actually spectrin and ankyrin are complementary. So spectrin is held together to the held close to the RBC surface by the ankyrin only. So in either cases the shape of RBC changes. About the properties of RBC, they form Rolex. Okay, RBC is piled together and this is called as Rolex formation. Specific gravity of RBC is about 1.092 to 1.101. About the packed cell volume, it is the percentage occurrence of RBC in blood. We have discussed the hematocrit value. Suspension stability of RBC is that they are uniformly distributed in blood. Okay. Now we have a term called metabolic limit of RBCs. We are not going into deep as it is a revision video. The value is about 34 gram in 100 ml of blood. Now come to lifespan of RBC. About the lifespan of RBC, we know it has a lifespan of about 120 days and it is destroyed in the reticular endothelial system. And to determine the lifespan of RBC, we can, like, this is the RBC, you can label it with some radioactive substances like radioactive iron or radioactive chromium, and the radioactive loss is directly proportional to the destruction of RBC. Okay, the lifespan of RBC can be determined easily. About the destruction, it goes to the spleen. We know it is the graveyard of RBC, and mainly the red pulp. Spleen has two pulps, the red pulp and the white pulp. So in the red pulp of spleen, the trabeculae of the red pulp of spleen mainly have a diameter of 3 micrometer, and the RBC diameter is 7.2 micrometer, so it gets destructed. Splenic capillaries are very small, so RBC destruction occurs, and the fragments are cleaned by the Kupffer cells or the reticuloendothelial cells. Now, destroyed RBCs, they release some substances, like when RBC is destroyed. Destroyed RBCs release substances like iron, and in this iron, apoferritin combines to form ferritin, and like that the ferritin is stored and reused. It also releases globulins, which goes to the protein pool obviously, and the porphyrins. Porphyrins convert into bilirubin, and bilirubin is excreted out. Function of RBC is transport of O2 from lungs to tissue. So in form of oxyhemoglobin, we know from our 12 that 97% O2 goes by that. Transport of CO2 to carboxyhemoglobin in the form, 30% nearly CO2 goes by that. Buffering action in blood, that is the H plus concentration regulation. Okay, that is also the function of RBC. And blood group determination, we have blood group antigens, we can discuss it later. Increase in RBC count is called as polycythemia. Actually, polycythemia is the marginal increase. Okay, the marginal increase or the temporary increase in count is called as polycythemia. It is not permanent. It is of two types, like physiological polycythemia and pathological polycythemia. Okay, physiological, we can say that in birth the RBC destruction is more. Okay, so in birth we can see a jaundice called as physiological jaundice. Physiological jaundice or neonatal jaundice. Is seen in birth, so this is also a cause of polycythemia. And before puberty and after menopause it also occurs. In mountain inhibitants, due to hypoxia, the erythropoietin is released from the kidney, so RBC count increases. In exercise too, adrenaline causes the spleen to contract, which causes the RBC count to increase. Okay, in emotional conditions it may occur, or after meals we can see a marginal rise in RBC. Decrease in RBC count. Decrease in count, if it occurs in abnormal conditions, we can call it anemia, but due to physiological conditions like increase in barometric pressure, due to which the oxygen tension of blood increases, so count decreases. In sleep, decreases, the RBC count decreases slightly during the sleep, and in pregnancy too, due to hemodilation. About the pathological polycythemia, so these are the clinical correlations you must know. About the pathological polycythemia, we have primary polycythemia, secondary polycythemia. So there's a variety of causes like respiratory disorders like emphysema. Okay, this can cause secondary polycythemia. Congenital heart diseases or Allergy disease. Allergy disease is mainly a pulmonary artery, so pulmonary artery mainly gets disrupted and all. Carbon monoxide poisoning may cause this too. Okay. Now we can have the shape variation in RBC, shape variations. Shape variations like RBC may get crenated, and crenation occurs in hypertonic solution. Hypertonic solution. You can have spherocytosis. You can have elliptocytosis. You can have poikilocytosis. One must note that in poikilocytosis, we have two specific shapes of RBC. Like they may become flask shaped, or hammer shaped. Okay. In structural variations of RBC, like some abnormal constituents are present in RBC. Okay. Like an example is lead poisoning. Lead poisoning. Due to which the RBCs look striated due to accumulation of basophilic materials. Okay. And like porphyrins are mainly accumulated in RBC. So this phenomenon is called as punctate basophilism. So you must know about the punctate basophilism. Some rings are mainly in abnormal conditions. Some ring may be present in RBC. Like this is the RBC, so don't mind the shape. These are the rings which are present and called as goblet rings. Okay, you can discuss about goblet rings. So goblet rings are mainly the twisted strands of basophilic materials. These are also basophilic. Okay, and some bodies may be found in abnormal conditions which are called as Howell Jolly bodies. In Howell Jolly bodies, these are the mainly nuclear fragments. Okay. Nuclear fragments accumulate in RBCs in the ectoplasm mainly of RBC. So about the Howell Jolly bodies you can look. Okay. So this was about the discussion mainly about the RBC. Let's start about hematopoiesis. We can mainly discuss erythropoiesis. Okay. Let's discuss Let's discuss erythropoiesis. It is an important question. So erythropoiesis is important. About the site of erythropoiesis in babies, children and adults up to age of 20. Erythropoiesis occurs in red bone marrow of all bones. Okay. After age of 20, erythropoiesis occurs in the membranous bones which are ossified by membrane. Okay, membranous bones like the vertebrae, sternum, ribs, scapula, skull. Okay, and the ends of long bones, the epiphysis mainly. You must have heard about the fusion of epiphyseal plate and the retention of red blood marrow. Okay, about that. So we have to discuss a little bit about the stem cells too. Let's discuss about stem cells. So in stem cells we have firstly some uncommitted. Uncommitted means the fate is uncertain. Okay, the fate is uncertain. Pluripotent stem cells. This convert into committed pluripotent stem cells. This divide into lymphoid stem cells, I am writing in short, okay, and colony forming blastocytes. Lymphoid stem cells mainly produce lymphocytes, and natural killer cells, okay, we'll discuss it in immunity. And colony forming blastocytes differentiate into CFU-E, CFU-GM, and CFU-M. Okay, CFU-E form RBCs. CFU-GM forms, GM stands for granulocytes. Granulocytes you must know, neutrophils, basophils, and eosinophils. And M is monocyte. And CFU-M form megakaryocytes. Megakaryocytes, which eventually fragment to form the platelets. Okay. So these are about the some little bit about the stem cells. We must have a basic idea of the stem cells. Okay. Okay. Like in mitosis and meiosis, we had learned that what are the changes that occur during mitosis and meiosis. Like in erythropoiesis too, we have few changes in RBCs during erythropoiesis. I will write erythropoiesis as EP. Okay, you must understand it. Changes in RBCs during erythropoiesis, like first of all, the cell size reduces, and nuclei and nucleus disappear, hemoglobin appears, and the staining property. This is an important point, that staining property of cytoplasm changes. Okay, we will see in subsequent steps. These are during erythropoiesis. Okay, before erythropoiesis, the RBCs are not there, so how will the changes occur? So some are the stages. These are the stages. First of all, we have the proerythroblast. Or we can say it as megaloblast. If you look at the structure, so you can see the nucleus is very large. Okay, large nucleus you can see, and 2 to 3 nuclei are found. Nucleoli, sorry, are found. 2 to 3 nucleoli. Hemoglobin has not appeared still, and the cytoplasm is still basophilic. So the proerythroblast converts into early normoblast or basophilic erythroblast. In this stage, the major event is that nucleoli disappear, and the color is still blue due to basophilic stain. Color is blue. Then it converts into intermediate normoblast or the polychromatic erythroblast. In this first time, hemoglobin starts appearing, HP starts appearing. Okay, so it is called as polychromatic because you can find both red and blue colors in that if you look through the microscope. Then the intermediate normoblast converts into the late normoblast. The late normoblast, also called as the orthochromatic erythroblast. Okay, Now it is majorly acidophilic. Acidophilic. And the nucleus is called as ink spot nucleus. Now, so ink spot nucleus is first time found in late normoblast. Now a procedure occurs which is called as the pycnosis. Pycnosis means the nucleus disappears, okay, ejects out of the late normoblast. So nuclear ejection is called as pycnosis. And the residue is called as reticulocyte. Reticulocyte is basophilic due to organelles that are destroyed, and then the remnants make it basophilic. Stains with supravital stain. Okay, a stain is specially used for it. It is called as supravital stain. Is used to stain the reticulocyte. And cytoplasm contains the reticulum, and reticulocyte is the immature RBC. It is the immature RBC. Okay. Reticulocyte within 1-2 days, 2-3 days, you can say 2-3 days, it enters into the capillary, and forms the mature RBC. Reticulocyte is so called because it has a reticular network. Okay, and in mature RBC the reticular network disappears. So the timeline is like that the proerythroblast till the reticulocyte stage, you can mention the timeline here. From proerythroblast till the reticulocyte stage, it takes 5 days. It takes 5 days. And from the reticulocyte to the RBC stage, you can take a average of 2 days. Okay, you must remember the timeline. Now we have the factors necessary for erythropoiesis too. We can discuss it. Factors necessary for erythropoiesis. So in case of hypoxia, we have the most important factor that is released from the kidney. It's called as erythropoietin. Erythropoietin mainly, in hypoxia, that is the low partial pressure of oxygen, the hypoxia inhibiting factor is produced, which activates the transcription and it produces erythropoietin. Okay, erythropoietin is a hormone released from the kidney. And stimulatory factors of erythropoiesis, we have thyroxine, erythropoietin first is, then second is thyroxine. Testosterone has some mild erythropoietic action. Then we have interleukins and a factor called a steal factor. You must pay attention to the steal factor. This is a stem cell factor. Okay, this is a stem cell factor. So these are the factors necessary for erythropoiesis, and these are all stimulatory factor. They stimulate erythropoiesis. Some are maturation factors of erythropoiesis. Okay, some are maturation factors. Like we have vitamin B12, cobalamin, or you can say cyanocobalamin. Okay, it is the anti-pernicious factor. You must have read in vitamins. It is the anti-pernicious factor. Okay. Then we have the Cassell's intrinsic factor. which are produced by the parietal cells of gastric glands, and deficiency causes gastritis, ulcers, etc. Then we have folic acid. If folic acid is less, the DNA synthesis mainly reduces, and that means that is stopped. Okay, so the anemia causes megaloblastic anemia. Megaloblastic anemia is caused in deficiency of folic acid. Okay, and due to this, the RBCs increase in size, and splenic capillaries are 3 micrometers in diameter. Okay, so due to this, large cells, they are destructed in mass. Okay, then we have some factors for hemoglobin formation. Okay, factors for HP formation. So we have proteins, amino acids. We have iron for heme synthesis. Proteins and amino acids are mainly for globulin synthesis. Iron for heme synthesis. Cobalt, nickel, copper. These are for iron absorption from GIT. Okay. You must know these facts as per the hematopoiesis or the erythropoiesis is concerned. Okay, and now we are going to our next topic, that is hemoglobin and the iron metabolism. We are discussing now hemoglobin. So hemoglobin is mainly a chromoprotein. It is a conjugated chromoprotein because it is of red color. Okay, its molecular weight is around 68,000 daltons. It is iron containing, and hemoglobin contained in birth. Okay, in birth, the hemoglobin is 25 gram per deciliter, and as per age progresses, after 1 year, we have 17 gram per deciliter, and at puberty and onwards in adulthood, so have 14 to 16 gram per deciliter. Functions: transport of gases and buffer action. So we didn't discuss it. If hemoglobin is present in plasma, okay, the viscosity of plasma will increase, osmotic pressure will increase, and it will be rapidly excreted out. Okay, so let's discuss about the structure of hemoglobin. Structure. So in structure, we have heme part and the globin part. Globin part is the protein part. It has 4 polypeptide chains, 2 alpha, 2 beta in case of adults, 2 gamma in case of fetus. And heme is the iron containing part. It is the pigment part. And it has 4 pyrrole rings, so it is called tetrapyrrole. Okay, and these are attached by CH4 bridges. You can see the structure from the book. And 4 pyrrole rings, you can name it as 1, 2, 3, 4 in Roman numerals and all. Okay. Types of hemoglobin: adult hemoglobin, it's HbA. HbA, it has 2 alpha and 2 beta. And fetal hemoglobin, HbF. One thing that is worth discussing is the hemoglobin saturation curve. Okay, so you may be asked in exam that which is fetal and which is adult hemoglobin. So this is adult, this is fetal. This is important because you may heard of a term called left shift. So fetal hemoglobin has more affinity for oxygen. So that's why the left shift has occurred from the usual adult hemoglobin. This is a sigmoid curve. Must have heard it. So abnormal hemoglobins. Now we have to discuss about the abnormal hemoglobins. Okay. Abnormal hemoglobin. So abnormal hemoglobin can be seen in case of hemoglobinopathies. Hemoglobinopathies. Like we can have HbS, sickle cell hemoglobin, in which valine is substituted for glutamic acid at sixth position of beta globin chain. This is HbS. We have HbC, which is called as hemoglobin C disease. This is worth remembering because you might have not heard about that. So we have HbC. In this, lysine is substituted in place of glutamic acid at sixth position of beta globin chain. Okay. So this is HbS and HbC. Now we have HbE2. HbE2, which is called as hemoglobin E disease. Okay. And we have HbS, HbH, which is called as hemoglobin H disease. Hemoglobin H disease, or also called as alpha thalassemia. In which the synthesis of alpha chain is decreased or absent or abnormal. And we have HbM2. HbM stands for methemoglobin. Methemoglobin is produced when there is a deficiency of cytochrome B5 reductase deficiency produces methemoglobin, and due to this HbM, the syndrome is called as blue baby syndrome. Okay, blue baby syndrome is caused where Hb is form of methemoglobin. Some other hemoglobins are produced in rare disorders, like we have contact spring hemoglobin, we have HbG, we have HbI, HbI, we have Bart's, we have Lepore, we have Kenya. These are the types of hemoglobins only. These are abnormal hemoglobins found in rare disorders. Okay. So abnormal hemoglobin derivatives are carboxyhemoglobin, where carbon monoxide combines with hemoglobin. We know that the affinity is 200 times more for CO. And we have methemoglobin. So methemoglobin is formed when iron Fe2 plus is converted to Fe3 plus. This is oxidized. So to prevent it, we have the NADH system. We may discuss it later. Okay. We have sulfhemoglobin, where hemoglobin combines with H2S. We have some drugs like phenacetin, nitrates, or sulfonamides. Okay, some drugs are there, sulfonamide drug or phenacetin. Okay, so these are caused by drugs. Okay, now let's see about the hemoglobin structure we have discussed. You can see the structure from the book, and Dissociation curve, about the dissociation curve, the right shift and the left shift. Okay, so let's continue. Right shift and left shift. So in this we can write about the factors that cause right shift or left shift. So right shift is caused by decrease in partial pressure of O2, temperature increase, increase in 2,3 diphosphoglyceric acid, DPGA, increase in CO2, decrease in pH, or increase in H plus concentration. Left shift is occurred in case of PO2 increase, temperature decrease, decrease in 2,3 DPGA, decrease CO2, increase pH. You must have idea about that. Okay, let's see about the hemoglobin synthesis. This is again a good question. HB synthesis. See, in hemoglobin synthesis, actually I have observed in Guyton, it has mainly simplified the process and completed it in 3-4 steps. So actually we are going to synthesize hemoglobin at the molecular level. Okay, so you can refer Guyton as and when you like, because it is the standard book and gold standard, but we will discuss in molecular basis. Okay, so heme synthesis and globin synthesis will be discussed separately. Heme synthesis occurs in mitochondria and the globin synthesis occurs in ribosomes, and this starts from the pro-erythroblastic stage. Okay, but hemoglobin appears in the intermediate normoblastic stage, we have discussed earlier. So let's see, let's draw a mitochondria so that we can represent it. So let's assume this is the mitochondria. Here, so we are discussing the heme synthesis. First step is the combination of 2-succinyl coenzyme A with 2-glycine in the presence of enzyme ALA synthase. ALA, you must have known that amino levulinic acid. Okay, so in presence of ALA synthase, it converts into ALA. It comes out of the mitochondria, 2 molecules of ALA, converts in the presence of ALA dehydratase to form porphobilinogen. In presence of uroporphobilinogen, one synthase, it forms uroporphobilinogen 1. Then again, in presence of porphobilinogen 3, co-synthase, it forms uroporphobilinogen 3, and in presence of uroporphobilinogen decarboxylase, it forms porphyrinogen 3. So this last compound, coproporphyrinogen 3, enters into the mitochondria again. So here we have the perfo-renogen 3. In presence of the oxidase enzyme, it converts into proto-porphyrin 9, and again in presence of protoporphyrin oxidase, it converts into protoporphyrin 9. Okay, protoporphyrin 9. It combines with iron and the enzyme here is ferrochelatase. Chelation occurs mainly the ring formation or that, and it forms the heme. Okay, so this is the complex pathway, I know, but you may practice it. This is the biochemical basis of the hemoglobin synthesis. So in exam you may write the Ganon part, the Guyton part, sorry, because it is pretty simple. Okay, this is for your knowledge. So globin formation occurs in ribosome. Okay, and polypeptide chains are formed, alpha, beta, gamma, delta. The chain has 141 to 146 amino acids. Okay. Now come to iron metabolism. Iron metabolism, quantity of iron in body. Let's discuss a little bit about the iron metabolism. So iron in our body is about 4 gram. Okay, about 50 to 150 microgram per deciliter of blood. Okay, we get dietary iron, and like dietary iron can be divided into two parts. One is non-heme iron and heme iron. You can get non-heme iron from veggies, grains, cereals, and mainly the plant sources, the green leafy vegetables. Okay, these are not easily absorbed, remember that. And heme iron, we can get it from fish, meat, chicken. So non-veg items mainly, they contain heme iron. Okay, now we may discuss the destruction of hemoglobin. So as per destruction, heme and globin are separated from each other, and in heme to iron and porphyrin. Iron is stored as ferritin. Ferritin is the stored form of iron, remember that, and it is reutilized. Porphyrin, it converts into biliverdin, then bilirubin, then into urobilinogen, lastly into stercobilinogen. Stercobilin, we can say stercobilinogen, then forms stercobilin. Okay, I'm directly writing stercobilin. And stercobilin is responsible for the color of stool. Now let's discuss about the globin. Heme is completed, now globin, and globin is to recycle. Okay, globin is to recycle. So we are seeing that the destruction of hemoglobin, many things are recycled. Okay, only the stercobilin that is lost. Okay, now discuss about the iron absorption. Iron absorption mainly occurs by the small intestine. Okay, the enterocyte cells of the small intestine are responsible for that. Right, enterocytes. Do that. So Fe3 plus in presence of ferric reductase converts into Fe2 plus, and to this Fe2 plus, let's say this is Fe2 plus, and we have protein called ferropoietin, and these are transported into blood. Actually this forms the Fe3 plus again, and this gets transported into blood. It combines with apotransferrin and forms transferrin. Okay, we can you can read the details in iron metabolism. Okay. About the storage of Fe, we have seen that it is stored in ferritin form, majorly ferritin, and some quantity is also stored in the hemosiderin form. Okay, less quantity, but ferritin is more. Stored in the reticular endothelial cells and in the hepatocytes too. About the daily loss of iron, in feces, 1 milligram of iron is lost. 1 mg of iron is lost daily via feces. Okay, and iron content of males is more than females. Okay, because in females in menstrual cycle the 50 ml of blood loss, so 25 milligram of iron is lost. Okay, then regulation of total iron in the body. So if feedback loop is formed and we have some factors that reduce the iron absorption, that is the apoferritin formation stops in liver, and transferrin gets completely saturated with iron. So these are some factors that reduce the iron absorption. Now we have come to the one of the most important topics, which are the blood indices. Blood indices. Okay. First is the erythrocyte sedimentation rate, ESR, also called as SED rate, also called as Bernanke reaction. Okay. So to determine ESR, we have two techniques. One is the Westergren method and one is the Winthrop method. In Westergren method, we take a pipette, mainly the tube that is of 300 mm, and 1.6 ml of blood is added with 0.4 ml of 3.8% sodium citrate, which is mainly the anticoagulant. Okay, so blood and anticoagulant is mainly 4:1 in ratio. This ratio is Westergren ratio. Remember it. In Winthrop method, we use a special pipette called as Pasteur pipette. Okay, Pasteur pipette is used, and 1 ml blood is combined with EDTA, which is the anticoagulant, and Winthrop's tube is placed in Winthrop stand, and 1 hour after 1 hour the reading is observed. Okay, so ESR is helpful to assess the progress of disorders like progress in disorders like we have 4 disorders for that. One is the tuberculosis, the pulmonary type. One is the rheumatoid arthritis. Again, one is the polymyalgia rheumatica, and fourth is the temporal arteritis. Temporal means the head region, so arteries get inflamed in the head region, so it is the temporal arteritis. Factors which increase the ESR and factors which decrease the ESR. So we have factors with increase ESR and decrease ESR. So which increase ESR is the specific gravity of RBC, increase the erythrocyte sedimentation rate because the viscosity is increased. So specific gravity of RBCs, Rolex formation, increase in size of RBC, decrease in number of RBC. Because in decrease in number the settlement is faster. Example, in anemia ESR increases. Okay, example is anemia. Then decrease ESR is the viscosity of blood. As viscosity increases, the settlement speed is decreased, so ESR decreases. And increase in number of RBC, ESR decreases. In physiological variations, as age increases, okay, age increases, ESR2 increases. In females, the ESR is slightly more as compared to males. ESR increases during menstruation. In pregnancy too, ESR increases. Okay, from third month to paturation. Okay, timeline is not so important. Pathological variations in ESR, like some diseases are there, like TB, anemia, tumors, rheumatoid arthritis, liver disorders. In all of this, ESR increase. Allergies, sickle cell anemia, leukocytosis, peptone shock, ESR decreases. Okay. Now we discuss about the PCV2 hematocrit value. Okay. So PCV determination help in diagnosis and treatment of anemia. Okay, decision of blood transfusion, like you must have remembered the normal PCV value too. Normal PCV values in case of males and in case of females. In case of males, the normal PCV value ranges from 40 to 45% of blood. Is the percentage of RBC in blood, okay, proportion of blood occupied by RBC. So in males it is 40 to 45%, and in females it is 38 to 42%. And increase in PCV occurs in polycythemia, in dehydration, because hemodilation occurs, okay, in dengue shock syndrome, dengue shock syndrome. Actually, dengue is of varying severity. In third and fourth severity, grade third and grade fourth severity of dengue, dengue shock syndrome occurs, which increases the PCV. Okay. Now come to decrease in PCV, occurs in anemia, liver cirrhosis, pregnancy, and due to ectopic pregnancy, vaginal bleeding occurs. So this also causes decrease in PCV. Now we'll discuss the blood indices. We have discussed the ESR and the PCV. Now come to blood indices. We have MCV, mean corpuscular volume, MCH, mean corpuscular hemoglobin, MCHC, mean corpuscular hemoglobin concentration, and CI, that is the color index. Okay. So let's say we will go not by the definition approach but by a example. Okay, so we cover it in least possible time. Let's say we have been given a blood sample where the RBC count is 4 millions per cubic mm. The hemoglobin concentration content is 8 gram per deciliter and PCV is given 30%. You must have observed till now that all are less. Okay. Let's talk about the color index first. To calculate color index, you must know the hemoglobin percentage, that is the hemoglobin concentration in subject, in patient, by hemoglobin content in normal person. Okay. Into 100. That is 8 by 15 into 100. That is 50. 15. So there we go. 3 and 20. Okay. So it will come around 53.3%. Okay. And the RBC percentage is RBC count in subject. By normal RBC count into 100. So this is pretty easy. 4 by 5 into 100, 20, 80%. Okay. Okay. So color index, the formula is HB percentage by RBC percentage. That is 53.3 by 80%. So I'm using the calculator. You can calculate it. This will be around 53, 0.67. Okay. This is the ratio. So it doesn't have any unit. Okay. The normal color index is 0.8 to 1.2. Color index increases. Okay, increase in color index occurs in anemias and decrease occurs in iron deficiency anemia. Increase occurs in pernicious, megaloblastic, and etc. anemia, and decrease occurs in iron deficiency anemia. So the subject is diagnosed with iron deficiency anemia. Okay. Now let's see the MCV. Color index is done. Now we'll calculate the MCV. So MCV is PCV in ml per 1000 ml of blood by RBC count in million per cubic mm of blood. Okay. So PCV count is given. You can write this formula in form of PCV in 1000 ml or in 100 ml into 10. Okay, PCV in 100 ml into 10. So you are given the PCV as 30 here, 30%. We multiply with 10 and divide by 4, as the RBC count is given 4. So this is around 2 and 75 cubic micrometer. Okay. Then comes the mean corpuscular hemoglobin. Okay, MCH. So MCH is hemoglobin in gram per 100 ml of blood into 10 by RBC count in million per cubic mm. Okay, so this is 8 into 10 by 4 and this is 20 picogram. Please remember the unit. Picogram is the unit. Okay. Okay, now comes the MCHC, mean corpuscular hemoglobin concentration. So this is hemoglobin in gram per 100 ml of blood by PCV in 100 ml of blood into 100. This will be 8 by 30 into 100. It will be 800 by 30, so this will be around 26.67%. Okay. Let's see at the normal ranges. So color index, I have told that the normal range is 0.8 to 1.2. This is the normal range. And MCV, the normal range ranges from the 78 to 90 cubic meter. This is the normal range. So we have 75, so this is less. This is the normal MCV. Now let's say the normal MCH, this is around 27 to 32 picogram. Okay, this is the normal MCH. So MCH is also low. Now MCHC, the normal range is 30 to 38%. This is the normal MCHC. So MCHC is also less. So that the patient suffers from microcytic because the size of RBC is also less, hypochromic because the quantity of hemoglobin is less, anemia, which is caused by iron deficiency. Okay, this is the preliminary diagnosis. So we have completed the blood indices too, and now we can discuss about the anemia too. Okay, let's discuss fragility first, then we will come to anemia. So we will discuss the hemolysis and fragility of RBC. So RBCs can be hemolyzed in normal conditions by the spleen after 120 days of lifespan. We have seen the process earlier, but hemolysis can occur in abnormal conditions too. Abnormal hemolysis is caused by various factors like hemolysins, abnormal shapes, some diseases, and few mechanical factors. Okay, this causes the abnormal hemolysis. Hemolysins are the hemolytic agents that destroy RBC by mainly by forming the pores. Okay, some pores are formed due to which the contents leak out and hemolysis occurs. Okay, so many hemolysins are there like phospholipase A2. Some examples can write like phospholipase A2. Actually, this is present in the snake venom. Okay, some chemicals like alcohols, benzene, CHCl3, some resins, arsenic. Some bacteria also cause that, like Clostridium tetani and Listeria species, some Listeria species bacteria. Okay, these are the hemolysins. And abnormal shapes occur in sickle cell anemia, thalassemia, hereditary spherocytosis, elliptocytosis. Okay, this need not be discussed. In diseases like autoimmune disorders, in some infections, some medicines or drugs to cause it, like some drugs are like cephalosporins, NSAID drugs, You know NSAIDs, that is non-steroidal anti-inflammatory drugs. Okay, so NSAIDs and penicillin, etc. Mechanical factors also cause hemolysis. Some mechanical factors you can write up, like can freeze the blood to cause hemolysis. You can you can give stress physically. Okay, like excessive exercising, severe exercise. So physical stresses, freezing blood. In bedridden patients, a machine is used that is called a heart-lung bypass machine. So, if a patient is in this machine too, so hemolysis may occur. Okay. Now let's discuss about the fragility. Fragility of RBCs. So, in fragility of RBCs, we have two types of fragility. One is the mechanical fragility. One is the osmotic fragility. Mechanical fragility is caused due to the wound or injuries, okay, and osmotic fragility is due to exosmosis, endosmosis. Okay, we can perform the fragility test. We can put the RBC in hypertonic saline solution and see their resistance. Okay, we can do a gradient test, like we can add NaCl from 0.2% to 1.2% taken in Corn's tube. So blood is added to it and mixed and left undisturbed. We can see the fragility, of course. Okay. Okay. So in Guyton, a specific thing is given that oxidative stress may cause hemolysis. Oxidative stress is mainly caused by the enzyme deficiency G6PD, okay, which maintains the level of antioxidant glutathione. It maintains glutathione levels. So in the deficit, it causes oxidative stress and hemolysis. Okay. So we have come to the end, the last topic of this lecture. We'll cover the WBCs and platelets later. Okay. So last topic and the most important topic, I'll say, that is anemia. So a variety of anemias, some classified otherwise, or I'll just write their names and describe it. Okay. One is the morphological classification of anemia, like we say microcytic, normochromic anemia. We say normocytic normochromic anemia and microcytic hypochromic anemia, all that. And we have the etiological classification, like hemorrhagic anemia, hemolytic anemia, aplastic anemia. Okay, so we'll discuss not about the classification, we will discuss mainly about the anemias, writing it. One is the iron deficiency. It is the most common anemia. Please remember, it is the most common anemia. RBCs are microcytic hypochromic. Okay. The symptoms are mainly same: the shortness of breath, the fatigue, tachycardia, okay, dizziness. Here, the MCV is less than 90 femtoliter. Okay, the MCV is less than 90 femtoliter. So microcytosis occur, obviously, you can see that. Causes, we can write like blood loss and low iron diet, poor absorption of iron from intestine. We have its features: the characteristic features I will only write, and the features like they're common. Okay, we can see brittle nails. Brittle nails. We can see a syndrome called as koilonkia. Koilonkia. In this, the nails become spoon shaped nails. Okay, you must remember this. And you can see also atrophy of tongue papillae. Atrophy of tongue papilla. The papillae in tongue, they get atropized and all. So this is the iron deficiency anemia. This is all about iron deficiency anemia. Now we have the pernicious anemia. Pernicious anemia, which is also called as addictions anemia. This is due to the deficiency of vitamin B12, the cobalamin, and the folic acid. B12 and folic acid deficiency, due to which the DNA maturation and condensation is not present. Okay, the RBCs become huge and macrocytic. They may be normochromic or maybe hypochromic, depending upon the cases. So normochromic is most commonly found. Macrocytic, normochromic, or hypochromic anemia. These are the status of RBCs. Let's discuss about the characteristic features. So features are lemon yellow skin. You can see a different type of tint in skin, that is lemon yellow skin, and red sore tongue. These are the two features of the pernicious anemia, okay, or the addictions anemia. Now we have the we can discuss, like pernicious anemia can be primarily autoimmune. Okay, in thyroid disorders and addictions disease, it mainly associated with that, so it may be autoimmune. Some neurological manifestations may occur, like paresthesia. Paresthesia is about abnormal sensations. Okay, paresthesia. We can see weakness. We can see ataxia. Ataxia is the gait changes or the muscular incoordination. Okay, these are the neurological manifestations. Cure is pretty simple. We can give an IM injection of B12. Okay, now let's discuss about the hereditary spherocytosis. If you set the RBC surface, we have, here is the upper lip of the RBC surface. Here we have the spectrin. And here we have the ankyrin, and some other proteins are also present here. These are miscellaneous proteins like glycophorins, band 3, protein 4.1. Okay, so to detect hereditary spherocytosis, we have a specific test called Coombs test. Okay, Coombs test is used for to detect hereditary spherocytosis. So in this, we know RBC becomes the globular shape, globular or spherical RBCs. This gets stuck in the splenic capillaries, and the resultant condition is called as the splenomegaly. or the hepatosplenomegaly, you can say. So splenomegaly occurs in hereditary spherocytosis. This is mainly genetic in origin. Okay. Now we have the glucose 6-phosphate dehydrogenase deficiency anemia. What I said, G6PDH. We discussed about it this in fragility, like deficiency anemia. Here, the glucose 6 phosphatase dehydrogenase phosphatase dehydrogenase decreases, due to which the glucose gets converted, due to which the glucose can't get converted, okay, into 6-phosphoglucolactone, and in turn the NADPH is decreased. Okay, you may think in that sense we will not go into the molecular details. Like you can say that the glucose is converted into pyruvate, so the glucose get converted into ribose 5-phosphate, and we have the pathway. Okay, so the hemoglobin, actually we can discuss the neurological manifestation, biochemical manifestations in biochemistry. So hemoglobin, this is, let's say this is the hemoglobin, and some reactive oxygen species, they bind over the hemoglobin. This is the ROS, reactive oxygen species, and let's say this is the RBC membrane, the inner leaf of the RBC membrane. This gets stuck here, this get stuck here, and here there is the hemoglobin, and these are called as Heinz bodies, which decrease the flexibility of RBCs. This reactive oxygen species actually accumulate due to decreased NADPH. Okay, and glutathiones, they react with the reduced form of glutathione, okay, GSH, and form a disulfide bond, and these all are the biochemical manifestations, so I am not going into deep. Then we have sickle cell anemia. Okay, we have sickle cell anemia. Or HBSS disease, or HBS disease, or sickle cell disease. This is mainly caused due to the missense mutation. Missense mutation. You must know what a missense mutation is. Otherwise, I will discuss it in genetics part. Okay. So here we know the complication, that what happens exactly, the valine and glutamic acid 6th position, beta globin chain. Okay. So hemoglobin molecules actually start polymerizing. Like this is the hemoglobin molecule, so they start polymerizing like that. They form a polymer, and the RBC. So this is the RBC. Due to polymerization of HB, accumulate as here, like that. This is sickling or sickling of RBC. Okay. We can see the complications, like due to sickle cell anemia, various complications may arise. And the complications are like jaundice. Okay. Jaundice. Due to blood supply occlusion, okay, we can see infarction. Hand and foot syndrome is usually talked with that. So this is useful for USMLE, of course. Okay, so we'll not discuss more. Vaso-occlusive crisis. We can hear about it. We have already heard, or we may not have heard about Trapezium. Trapezium is the erectile dysfunction of the genital organs of males, okay, due to the blood and abnormal erection. Then we can see splenomegaly, obviously due to the tricentric shape and the odd shape. You must have heard that sickle cell anemia patients have resistance to Plasmodium falciparum too. Okay. Lastly, we'll discuss about the we have more anemias to discuss. Obviously, we'll discuss about the hemorrhagic anemia. Hemorrhagic anemia is due to the blood loss. It is pretty simple. So in hemorrhagic anemia, the RBCs are microcytic, hypochromic. Then we can discuss about the aplastic anemia. Here what happens, a term is prevalent, that is pancytopenia. Pancytopenia. Due to this pancytopenia, the RBCs, WBCs and platelets, they decrease in number. RBC, WBC and platelets, this decrease in number. Okay, this is called as pancytopenia. Actually, in aplastic anemia, 50% of cases the cause is unknown. Okay, so it is also called as idiopathic. It is called as idiopathic because the cause is unknown. We have the reasons like RBC, they are normocytic, normochromic. RBCs are mainly normal, you can say. Okay, this is due to red bone marrow destruction. Aplastic anemia, the cause is red bone marrow destruction and replacement of it by yellow bone marrow. Okay, reasons are many, like X-rays, bacterial toxins, and lupus erythematosus. Okay, you can discuss it completely. Thalassemia. About the thalassemia, we saw thalassemia. It is also called as Cooley's. Sorry, spelling is wrong. Cooley's anemia. You can call it Mediterranean anemia. Okay, because it is common in Thailand and Mediterranean ancestry mainly. MCB is less, so MCB is less than 90 liters, FM to liter, sorry. So it is a microcytic anemia. Thalassemia major and minor are two types. Thalassemia major, thalassemia minor are two types. Major is less common and minor is more common. And this is the major, less common, but the severest form, severe form, and this is the mild form. Okay, so alpha thalassemia and beta thalassemia are two classifications. In this alpha thalassemia, 1 alpha plus 2 beta are found because the alpha globin chain decreases in number. In beta thalassemia, 2 alpha plus 1 beta. Okay, this is the most common type. Beta thalassemia is more common. Cure is bone stem cell transplant and transfusions. Now we can discuss about the hemolytic anemia. Hemolytic anemia, this due to hemolysis or RBC destruction. Okay, common causes like liver failure, renal disorders, okay, hypersplenism, and infections like hepatitis, septicemia. Hemolysis, hemolytic disease of newborn, HDN, may cause it. Okay, autoimmune disorders. Actually, due to this, the fragile RBCs are produced. Okay, fragile RBCs produced. Due to protein deficiency, anemia is also caused, in which the RBCs are macrocytic because due to protein deficiency, the size of RBC increases, and hypochromic. Megaloblastic anemia, we saw it earlier, that is the pernicious anemia. So deficiency of folic acid. Due to a folate trap is also discussed in that. This is the complementary deficiency in folic acid and vitamin B12. Okay. Then we have the anemia of chronic diseases. Like some chronic diseases are there, like we can have a disease called as Hodgkin's disease. You can have lung cancers. You can have chronic renal failures. You can have rheumatoid arthritis. You can have infections like tuberculosis, lung abscess. So in this all diseases too, this is the second most cause. Okay, this is the anemia of chronic diseases. This is the second common type of anemia. This is the second common type of anemia. So effects of anemia, like some effects are occurred. Last topic it is effects of anemia. The common effects majorly: splenomegaly, hepatomegaly, effects on digestion, digestion effects, like we have many of them, like anorexia, nausea, vomiting, constipation. You can see a stool called as black maroon. The color of the stool is black maroon stool. Okay. Indigestion, the papillary atrophy of tongue occurs in pernicious anemia. Okay. And necrotic lesions in aplastic anemia. So kidney, and majorly in kidney, is C. albuminuria. Albuminuria in kidney. You can see pale skin, brittle nails. Menstrual cycle is disturbed, so disturbed menses. In CVS effects, you can see 3-4 effects, like you can say tachycardia, cardiac output increases, cardiac murmurs. Okay. These are all effects of anemia. So in the next class, in the next revision session, so let's finish it because it has been a long class and we have a lot of content to cover and all. We have discussed about blood and about plasma proteins in detail, then erythropoiesis, the whole procedure, about the hemoglobin, iron metabolism, and about the blood indices like ESR and PCV, color index, MCV, MCH, MCHC. About the hemolysis and fragility of RBC, and lastly about all the types of anemia and the causes and the effects of anemia. So in the next topic we will take up in the next part of this we'll take off WBC's the immunity and about the blood coagulation. Many clinical features are there: blood coagulation and blood grouping, transfusion, all that. Okay. Hello guys, so we'll discuss about the WBCs now after RBCs, the white blood cells. This is also an important topic. So classification of WBCs is like granulocytes and agranulocytes. Granulocytes constitute of neutrophils, basophils, and eosinophils. And agranulocytes constitute of lymphocytes and monocytes. So neutrophils are majorly amoeboid in shape. They take up the take up both stains. In fact, neutrophils, because they are neutral, so they can take both stains. They can take acidic stain as well as basic stains. These are polymorphs. Okay, these are polymorphic and constitute 50 to 70% of total WBCs. Their nucleus is multi-lobed nucleus. Okay, younger cells have no lobe, but as the cell ages, the lobe starts appearing, 2 to 5 lobes, let's say. Okay, some granules are there, of course, granulocyte. So we can stain it with a stain called as the Leishman stain. Must have heard about the stain. Of course, you must have heard in hematology to see the peripheral blood smear. We use the stain. And about the basophils, these are the least abundant, that is 0 to 1%, and the diameter is 8 to 10 micrometer. They have coarser granules. Their granules are very coarse. So the nucleus, when you see in the peripheral blood smear, the nucleus is not seen because the granules are coarse. Okay, coarse granules. And the stain, they stain with the methylene blue, so they look purple. Okay, bilobe nucleus is present. Bilobe nucleus is present. The about the eosinophils. Okay, about the eosinophils, they have also coarser granules, and they are pinkish in color, slightly pinkish with eosin. The stain with eosin, it stains pinkish or oranges, you can say. The nucleus is has a specific name, which is called a spectacled shaped nuclei. Okay, nucleus, spectacle shaped nucleus. The abundance is about 2 to 4% of all WBCs. The nucleus is spectacle shaped, or you can write bilobe too. Okay. About the lymphocytes. Lymphocytes are classified into two types. One is the large lymphocytes, one is the small lymphocytes. Large lymphocytes are majorly 10 to 12 micrometer, and small lymphocytes are 7 to 10 micrometer. Based on function, these are classified into B lymphocytes and T lymphocytes. B lymphocytes provide the humoral immunity, and T lymphocytes are responsible for cell-mediated immunity or cellular immunity. And the lymphocytes are they don't have granules, and their abundance is 20 to 30% abundance. They don't have granules. So the nucleus occupies the major position, majority of the cell. Okay. Now comes monocyte. Just a minute. Okay. So lymphocyte is done. Then we have the monocytes, which are the largest of all leukocytes, largest leukocyte. They constitute 2 to 6% abundance, and the nucleus is maybe variable shapes, like round or maybe oval. Sometimes they are of bean shaped. Okay. Okay. So applied in the applied physiology part, we have leukocytosis, and we have leukopenia. Leukocytosis is the increase in WBC count, and leukopenia is the decrease. Okay, so we have variations in WBC count. Okay, variations. We have physiological variations as well as pathological. We have physiological variations. We have pathological variations. Like with age, Okay, the normal WBC count is the range of 4,000 to 11,000. We know that cubic mm. This is the normal range. You must remember it. And in infants, the count is 20,000, and children 10 to 15,000. Okay. According to sex, in males the WBC count is more than females. Diurnal variations, like in morning, WBC count is more as in afternoon. In afternoon, the WBC count is more as compared to morning. In exercise, it increases slightly. In sleep, it decreases. In anxiety, it increases. In pregnancy, it increases. In parturition, it increases. In menstruation, it increases. Okay, so host of pathological variations we have, then physiological variations. And pathological variations we have like leukopenia, leukocytosis, leukemia, like granulocytosis. Okay, so any other leukopenia, we first discuss leukopenia. Leukopenia is the decrease in WBC count, and the causes may be radiation exposure, drugs like chloramphenicol, thimerosal. Okay, so in leukopenia, there is the anaphylactic shock. We'll learn about this in allergy, of course. Liver cirrhosis, spleen disorders, viral infections. Okay. Next, in leukocytosis, it is increase. So in that, the manifestations are infections, allergy, common cold, TB, glandular fever. Next comes leukemia, which is called as the blood cancer. One is the acute and other one is the chronic. Okay, so these are of 4 types, you may say. Okay, these are of 4 types. One is the acute lymphocytic, one is the chronic lymphocytic. Okay, acute is rapid increase and chronic is gradual increase. And one is acute myeloid cells. What's are myeloid cells? Granulocyte and monocytes. These are myeloid cells. So leukemia, we have 4 types, I said: acute lymphocytic, chronic lymphocytic, acute myeloid, and chronic myeloid. Okay. Risk factors we have like same: radiation exposure to chemicals like HCHO, benzene, smoking, genetic disorders like Down syndrome. Down syndrome may cause an incidence of leukemia, that is the blood cancer. Common signs and symptoms like skin discoloration, weight loss, tiredness, bruising, bleeding. Okay, these all may occur. So differential leukocyte variations, we have neutrophilia, we have neutropenia, eosinophilia, basophilia, and monocytosis. Okay, many are there. So we need not discuss it individually. You can see the book and about the lifespan of WBCs. This dependent on demand. These are dependent on demand and type. In tissues, generally they persist for months, and whereas in they may be destructed in 10 days or so. Okay. Some properties are there. Properties like one of them is diapedesis. I'm sure I have discussed this earlier, where narrow blood vessels, they can escape out or squeeze out. They show amoeboid movement. They show chemotaxis. Chemotaxis is the aggregation or the process by which the WBCs, they reach up to the site of injury and plug the blood. You can see that in chemotaxis in the blood coagulation part, hemostasis part. Then phagocytosis. Okay. Functions of WBC, neutrophils are the first line of defense. I haven't wrote it. This is the first line of defense. Okay. They release a variety of substances. The granules have enzymes. Okay. The granules have enzymes. So the functions of RBC we will discuss. Discussing the few important things of WBC. So I was talking about the functions of WBC. Okay. The functions of WBC. In that, we first have neutrophils. I said that it is a first line of defense, as we wrote earlier here. First line of defense. And the granules of neutrophils, the granules have some enzymes. Many enzymes are present in the neutrophilic granules. Like neutrophils have to destroy many microbes, bacteria, fungi. Okay, and they have some platelet activating factors too, like PAF, platelet activating factor. Okay, PAF actually is a cytokine which activates the platelets so that they accumulate at the site of injury, as we'll see in the hemostasis. Then NADPH oxidase. NADPH oxidase has primarily bactericidal applications. Okay, it kills the bacteria. Okay. Then we have defensins and cathelicidins. These are antimicrobial peptides. Remember that these are antimicrobial peptides. Okay. Then we have proteases, myeloperoxidases, elastases. Okay, we have proteases, myeloperoxidases, elastases. These are all for microbial destruction. Okay, these are all for microbial destruction. Now a term is that respiratory burst. Okay, respiratory burst is the rapid increase in consumption of O2. What happens in phagocytosis? We have the process of phagocytosis. In phagocytosis, the demand for O2 increases rapidly. Okay, demand for O2 increases rapidly. So respiratory bursts can occur. Okay. So after discussing respiratory burst, this occurs at the time of phagocytosis, and the neutrophil membrane, they produce the NADP oxidase enzyme present in the neutrophilic membrane produces some superoxides like ROS, free radicals. Okay, which have potent bactericidal applications too. So these are the pathophysiology of the respiratory burst. Okay. You must have heard the term pus. Pus. Okay. This is the whitish yellow fluid, and this constitutes of dead WBCs plus some bacteria trapped in it, serum, and some debris. Okay, these are the constituents of pus. Then we have eosinophils. The functions of eosinophils. Eosinophils provide defense against parasites. Okay, parasitic defense is major function of eosinophils. Okay. Okay. So eosinophils secrete some lethal substances like eosinophil peroxidases. Okay. Eosinophils secrete eosinophil peroxidases, a compound called MBP, sorry, major basic protein, ECP, eosinophil cationic protein, which is a neurotoxin, and EDN, eosinophil derived neurotoxin. Okay. So let's say to understand the action of MBP, this is very important. Let's say this is the helminth. Okay, let's say this is the helminth, the parasite or the helminth. So MBP reacts to it, and forms the distension or the ballooning of the outer wall of the helminth. Here's the outer wall. I'm shading with blue. So it causes the ballooning like that. Okay, it causes the ballooning, and this gets pouched off. So distension and detachment of tegmental sheath. This is the tegmental sheath of the helminth. You must have known the parts. Okay, and it causes the death. So this is one of the way in which MBP acts. Okay. Then we come to basophils. Basophils have important role in allergy. Okay, basophils have a role in allergy. Actually, the basophils have IgE receptors on them. Let's say this is the basophil. Okay, and they have some certain receptors on them. These are the IgE receptors. Okay, and the IgE antibodies, IgE immunoglobulin E antibodies bind to it and play a role in immunity. Okay, so basophils release a few substances we must know. One is the heparin to prevent the intravascular blood clotting. Intravascular blood clotting prevention. Second is the histamine, bradykinin, serotonin. Okay, next is the proteases and myeloperoxidases. which destroy the microbes, destroy microbes. Then some cytokines too, some cytokines too, like for example interleukin 4, IL4, IL4, it's not visible maybe. IL4, it accelerates the inflammatory reaction, accelerates inflammatory reactions. Okay, interleukin 4 accelerates the inflammatory reaction. Then comes mast cells. Okay, we'll discuss about a special cell called as the mast cell. Mast cell resembles basophils. Okay, but when the basophils actually lodged in the tissue spaces. Okay, let's say here is the cell, here is the cell, here is the cell. This is the tissue, and let's say a basophil is there here, so it gets lodged here, and the tissue basophils are called as mast cells. Okay, the lifespan increases when they lodge here. Okay, so their function in allergy and anaphylactic reactions, majorly in allergy, you can say. Okay, and the secrete substances, majorly basophils, the substances secreted by basophils are also secreted by the mast cells, plus some extra substances like chondroitin sulfates and proteoglycans. Okay, then mast cells produce leukotriene C2 and prostaglandins and cytokines. Okay. Next is the monocyte. Next is the monocytes. Monocytes are the largest cells. We have noted earlier that the precursors of tissue macrophages, as basophils, which lodge in tissue form mast cells. Like that, monocytes, when get lodged in the tissue, they form macrophages. Monocytes to constitute of the first line of defense. They are motile, they are phagocytic, they have phagocytic activity. Then tissue macrophages, monocytes get lodged in tissues from macrophages, and macrophages in body are present in various organs, like in liver they have different names. Liver, it is called as Kupffer cells or reticuloendothelial cells, of course, and in lungs they are called as alveolar macrophages. We have dust cells, we have splenic ones, splenic macrophages, etc. Various types. Monocytes secrete substances like IL, interleukin 1, MCSF, macrophage colony stimulating factor, and from PAF2, platelet adhering factor. So these are the substances secreted by monocytes. Lymphocytes will be discussed in the immunity part, because majorly T lymphocytes and B lymphocytes are the part of discussion in immunity. Okay, then we are discussing the topic called as the leukopoiesis. Leukopoiesis, first is the pluripotent stem cells, PSC. Okay, PSC converts into lymphoid stem cells. So the pluripotent stem cells get divided into the lymphoid stem cells, and the colony forming blastocytes. Okay, and we know that the colony forming blastocytes form CFU E, CFU GM, and again CFU M. The lymphoid stem cells form lymphoblasts, and again lymphocytes. The CFB or colony forming blastocytes, CFUE forms RBC, GM forms myeloblast. Myeloblasts. Myeloblasts again divide to form promyelocyte, and monoblast. Monoblast forms promonocyte, and this promonocyte converts into monocyte. Promyelocyte forms neutrophil myelocyte, basophil myelocyte, and eosinophil myelocyte. This neutrophil myelocytes form neutrophil metamyelocyte. I am writing it in short. And basophilic myelocyte forms basophil metamyelocyte, and eosinophilic myelocyte forms eosinophil metamyelocyte. This forms the neutrophil. This forms the basophils, and this the eosinophils or acidophils. Okay. So this all gets completed about the leukopoiesis. This is the general steps and phenomenon of the leukopoiesis. Now we are going to a very important topic called as immunity. Please listen very carefully about all the important things that you must remember in immunity. Okay. So immunity, that means that can be of many types. Like it may be of innate type, or it may be of acquired type or adaptive type. Okay. So we can divide immunity into innate immunity and acquired immunity. Innate immunity is inborn. Okay, it is inbornly present. It is the natural type. It is the non-specific type, not specific for any particular antigen or any particular microbe. It constitutes the first line of defense. Acquired immunity, meanwhile, maybe due to it may produce the resistance against the foreign bodies. It is the specific type, and it is the most powerful type. Again, the acquired immunity may be divided into two types. Maybe of cellular type or cell mediated immunity. It may be of humoral immunity. The examples of innate immunity are many, like digestive juices are the examples of innate immunity. The neutrophils, the lymphocytes, the macrophages, the natural killer cells, some antimicrobial peptides. You must have remembered the antimicrobial peptides, defensins and cathelicidins. These are the constituents of innate immunity. Acidity in specific organs like urine. Urine is acidic because it provides immunity. Okay. Stratum corneum in skin is a layer. Stratum corneum. In skin, stratum corneum is present as the innate immunity. It is the first line of defense. Okay, the skin and the mucosal membranes they constitute the first line of defense. So the innate immunity. In skin, another cell is there that is called as Langerhans cells. Okay, in skin, and many interferons are there. Interferons are basically proteins which are secreted with in response to a viral infection. Okay, to to aware the neighboring cells of the impending danger. Okay. So lymphocytes are mainly of B lymphocytes and T lymphocytes. We know that. Why we call it as B lymphocytes? Because it was first discovered in the bursa of Fabricius. It is the primary lymphoid organ in case of the aves, the birds. Okay. So we call it as B because B stands for bursa. Okay. And T stands for thymus because the lymphocyte migrates from the thymus, and in thymus, thymosins are released. secretes thymosins, which convert the premature T cells to mature T cells. Okay. We may have heard about the term called as antigens. Okay, antigens, because anti here stands for antibody, and the gens mean generator. Okay, so these are antibody generators. They generate antibodies against themselves. Okay, these are mainly proteins or polysaccharides. So its molecular weight must be greater than 8,000. Okay, we can describe the structure by a triangle. Antigens. Okay, they have epitopes. These angles are called as epitopes. These are the binding sites for antibodies. Okay, epitopes bind to antibodies. So let's discuss about the development of the T cells. T cell development. How the T cells develop. So the immature T cells, they first start the journey from the bone marrow. Okay, first start the journey from the bone marrow. From bone marrow, they migrate to the thymus gland. You must have heard about the thymus gland. It is a lobular gland. They come to the medulla portion of the thymus gland. This is the thymic medulla. This the lymphocytes come here. Let's say yellow color. And after that, let's zoom at the thymus gland. Let's zoom as the at the cortex and medulla of the thymus gland. That means I am zooming in to this space here. Okay, let's say this is the cortex, and this is the cortex, and this is the medulla. Okay, here the immature T cells, let's say this is the immature T cell. Okay, they gain a specific receptor on their outer surface, which is called as TCR. Okay, T cell receptors. TCR, the mature T cells. Now this is immature, this is mature. This is called sensitization. A term is used, sensitization. Sensitization means the T cells, they gain a specific receptor called as TCR. This come into the medulla. This mature T cells come into the medulla with the TCR. Okay, they may express two types of things. They may express a double positive cell or a double negative cell. Let's say this mature T lymphocyte, it adheres to CD4 and CD8. So we call it as double positive cell. But if it adheres with, let's say, CD25 and CD44, we call it as double negative cell. Okay, so double positive cell and double negative cell, this may undergo a process called as positive selection. This process is very important. That is called as positive selection. In the positive selection, it may develop into a single positive cell. It is now double positive. The CD4 and CD8 both are present. After positive selection, it may be of like this. One may be removed. It may be CD4 only, or it may be CD8 only. Okay. And here in double negative cell, it can attack only self cells. Okay, so it undergoes apoptosis. Okay, So we discussed about the development of the T cell, the double positive cell, the double negative cell, and the positive selection. Okay. Now let's come to the topic T cell types. Okay, T cell types of the T cells. So these are Th cells, T helper cells, or CD plus T cells. These are also called as CD plus T cells, and because CD4 is present at the surface. Okay. So we can call it a CD4 T cells or CD plus T cells. It may be of cytotoxic type, TK or TC. Here K stands for killer cells. Okay, these are T killer cells. This may be called as CD8 plus T cells. Why this is called as that? Because CD8 must be present in the surface. So here CD4 and the T cells having the CD4, these are the helper cells, helper T cells, and these are the killer T cells. Okay, and many types like suppressor T cells, TS cells, T suppressor cells, and memory T cells, TM, the memory T cells. Okay, so this is the whole phenomena in which we express the T cell types. Okay. I have not discussed about the beta selection, so here we go. The TCR, the mature T cells, when they come into the medulla, there is a specific process occurring at the interface of the cortex and the medulla, and this is called as beta selection. Beta selection is the process that is occurring at the interface of cortex and medulla. Okay. Now let's talk about B lymphocytes. B lymphocytes are processed in liver in case of fetus, or in bone marrow in adults. Fetus, bone marrow in adults. Okay. These are transformed into two types of cells. One is the plasma cell, and another one is the memory cell. Memory cells are stored, and plasma cells give rise to we will see in the later part the various immunoglobulins. Okay, they give rise to the IgA, IgM, IgD. Okay, now let's talk about the antigens. Antigens are mainly conjugated proteins. They may be lipoproteins, they may be nucleoproteins. Okay. Antigens are two types. One is the self antigen. Example of self antigen is RBC has its own antigen like A antigen and B antigen. Okay. One is the foreign antigen. On the basis of a response, antigens may be divided into antigens inducing antibody production or antigens producing allergic reaction. Okay. So foreign antigens may be classified into two types. One is that induce antibody production, and another is it may produce allergic reactions. Okay, so foreign antigens may be of like microbial toxins, some allergens, some transplanted material. You may heard of a term called as the graft rejection. Okay, graft rejection. Graft rejection is due to T cell mediated or cell mediated immunity. Okay, you may have heard of the important question in NEET times, of course. So cell mediated immunity, let's talk about the cell mediated immunity or the cellular immunity. First, we have to discuss about the specific type of cells called as the antigen presenting cells, APCs. Okay, antigen presenting cells. APCs we can see in three distinguishing types. One is the macrophages. Macrophages are the main APCs. Okay, these are the main APCs. One is the dendritic cells. Another is the B lymphocytes. B lymphocytes. Okay, these are the APCs. Dendritic cells, please remember, dendritic cells are non-phagocytic. Non-phagocytic. Dendritic cells may be present in various organs, like in spleen. Dendritic cells of spleen are there. Then lymph nodes have follicular dendritic cells. They may be present in lymph nodes too. They may be present in the Langerhans cells. Langerhans cells are basically the modifications of the dendritic cells only. Langerhans cells. Okay. And these B lymphocytes, they are of dual function. They are antigen presenting cell as well as antigen receiving cells. Okay. They receive the antigen too. So they are antigen receiving cells too. They are the dual cells. Okay. Okay. You might have heard about the word called as MHC. MHC. Major histocompatibility. Major histocompatibility complex. Major histocompatibility complex. This is exactly present in the short arm of chromosome number 6. Okay, chromosome number 6 has a short arm and it is present here. So MHC may be divided into two classes. MHC, it is one is the class 1 MHC. The second one is the class 2 MHC. Class 1 MHC is present on every cell. Present on every cell of the body. Okay, it presents the endogenous antigens. The antigens that are present in the body itself, the class 1 MHC express them. Class 2 MHCs are present on specific organs, like present on B cells, macrophages, and the antigen presenting cells. Okay, these three positions in which class 2 MHCs are present, and they present exogenous antigens. Present exogenous antigens, like antigens of bacteria, virus. These are exogenous antigens. Actually, what happens? The APCs, they engulf the exogenous, the bacteria, or the APCs engulf the bacteria or the toxins. Okay, so APCs, like let's say here is a bacteria with an antigen. This is the bacterial antigen. It is the bacteria, and here is a macrophage. So the bacteria is engulfed by the macrophage, and the macrophage separates the bacteria from the antigen. So here is the antigen, and then class 2 MHC is present over the macrophage, so it presents it here, and now antibodies can bind to destroy it. Okay, this is the whole scenario that is going to occur. Okay. Now we are discussing about the activation of helper T cells. Helper T cells get activated to complex reactions mainly. APCs have HLA and class 2 MHC, and the TH cells have TCR. So antigenic peptide gets activated by that. So T cell gets activated and provide immunity. Leaving that, we will discuss about the helper T cells. Okay. Helper T cells. About the helper T cells, these are of two types. One is the Th1 cells, and second one is the Th2 cells. Th1 cells secrete IL-2, B cell growth factor, and gamma interferons. Okay, these gamma interferons, they give positive feedback, or they stimulate the TC cells, the cytotoxic cells, macrophages, and the natural killer cells. Okay, and the Th2 cells, they provide humoral immunity, and they secrete IL-4 4, 5, 6 and 10. Okay, this help in B cell activation, antibody production, etc. Okay, so these are the things which you have to remember in the helper T cells. And now we come to the cytotoxic T cells. Cytotoxic T cells. These cells attack directly. Okay, so one let's say this is an organ, the transplanted organ that is not accepted by the body and seen as foreign. Okay, or foreign antigens. They come here. Here is the T cells, and this is the T cell receptor. Let's say this is the antigen. The antigen gets bind to it, and this releases lysosomal enzymes which destroy the T cell, destroy the antigen. So this gets fragmented or destroyed. Second one is the cancerous cells. Cancerous cells, might have heard about the malignancies, about the contact inhibition of the cancerous cells. Okay, about different things, the metastasis and all. So cancerous cells and transplanted organs, these are also destroyed by TC cells. And virus infected cells, it also kills the own cells. The virus infected cells are removed from the body by the help of cytotoxic T cells. Okay, now let's talk about the suppressor T cells. The suppressor T cells, or the regulatory T cells. The suppressor T cells or the regulatory T cells, this suppress the activity of T killer cells, suppress TK cell activity. It prevents the T killer cells to destroy its own cells. Okay, because if it starts destroying the own cells, then there will be a problem in the body. Again, we are discussing about the memory T cells. Memory T cells, they remain in lymphoid tissue and migrate when exposed to the same organism. Okay, they give positive feedback. They activate the T cells, TK cells and all, which destroy the antigen. Okay. Now let's talk about the humoral immunity. We discussed now about the cell mediated immunity. Now let's talk about the humoral immunity. About the humoral immunity, B cell, we told that the B cell, it produces antibodies which are gamma globulins, and these antibodies are present in blood and lymph. Actually, in older times, the blood, lymph and all were called as humors. Okay, HUMORS, humors. So the name is that humoral immunity. Okay. So B cell activation, how does the activation occurs? Activation of B cells. So let's say this is an antigen presenting cell. It must have an antigen here. Here is the B cell. Here is the BCR, B cell receptor. Okay, the BCR. So due to this interaction, the antigen presenting cells secrete interleukin 1, IL-1. This IL-1 activates the B cell, activates B cells. Now the B cells gets activated, they proliferate, they differentiate into the plasma cell, which secrete the antibodies. Antibodies move to lymph and get circulated. Then the another proliferated form is the memory cell. It is inactive till next exposure of pathogen. and in case of second exposure it stimulates the antigen to form antibody. Okay, now we will talk about the antibodies. Let's talk about the antibodies. Antibodies are also called as gamma globulins. Okay, these are the gamma globulins or the immunoglobulins. You might have seen the structure of the antibody in your previous classes and all. So we will not discuss more. Okay. This here and this, the red shaded part, are called as constant regions, and the blank part are called as variable regions. So this is the constant region. Okay, and these are the variable regions. You have the heavy chain and the light chain and all. We have some disulfide bonds to here, like disulfide bonds are present. Okay, this is called as VH, this is called as VL, this is VH again, this is VL, and this is called as CH. Okay, CH2, CH3, etc. This region here is called as the hinge region. This whole region here is called as the hinge region. Okay. You might have read the structure in detail too in previous classes. Okay, so we have the regions. We saw that we have some constant regions which are FC and some variable regions which are FAB. Okay, where C stands for crystallizable fragment. Crystallizable fragment. Where C doesn't stand for constant. Okay, C stands for crystallizable fragment. And FAB are the antigen binding region. Okay, this FC crystallizable fragment causes complement fixation. Complement fixation. Complement fixation. Now let's talk about the different antibodies. We have IG, G, IG, A, IG, M, IgD, and IgE. Okay, this is gamma day, and this is in the order of abundance. Okay, IgG is the most abundant, IgE is the least abundant. IgA is also called as a secretory antibody because present in the body secretions like in colostrum and all. It provides localized defense. IgA is the secretory antibody. The secretory antibody. Okay, the IgG, it helps in complement fixation. It helps in complement fixation. I am writing one of the functions only. You can read the functions from the book. IgM also helps in complement fixation. IgE helps in allergic reactions, and IgD helps in antigen recognition. Okay, so these are the things which you have to remember. So action of antibodies, we will now discuss action of antibodies. Antibody action. So we have two types of action. We have a direct action, we have an indirect action. Direct action, indirect action, first what will occur? First agglutination will occur. The antibodies and the foreign antigens are clumped, right? And then precipitation will occur. Soluble antigens are converted into insoluble PPT. Okay, then neutralization will occur where toxic sites are covered by the antibody, and lastly lysis will occur. Lysis in which the cell membrane of the foreign organism ruptures and the invader is destroyed. Okay. Now we come to indirect action. Indirect action often functions by the complement system to complement system. These are the system of plasma enzymes. Okay, complement system you might have not heard. It is the system of plasma enzymes. About 11 plasma enzymes are present. Okay, this forms some like C1, some complement proteins like C1, C2 up to C9 are there. These are inactive, of course. And then the C1 may be distinguished into three types in the C1P, okay, C1Q, C1R. These are the complement proteins. Okay, so 11 are there. Now indirect action is needs activation. Okay, it is more strong as compared to direct action. Indirect action is more strong and activated by three pathways. One is the classical pathway, another is the lectin pathway, and the third is the alternate pathway, or the you can say by complement protein alternate. So we'll take an alternate pathway too. By classical pathway, the C1 binds with the antibody. It causes opsonization of neutrophils. It stimulates the neutrophils and the macrophages, and this is called the lysis of the cell membrane, lysis of cell membrane. Okay. After lysis, they attach the leukocytes to the site. We may write it chemotaxis. It attracts the leukocytes to the site, cause agglutination, then cause neutralization, then histamine is released, and lastly these are destroyed. Okay, the foreign invader is destroyed. Lectin pathway, so the pathogen here in the cell membrane, let's say this is the pathogen in the cell membrane. Let's take a section of the cell membrane here. So here's the cell membrane section. We have some fructose and mannose groups, of course, in the cell membrane. About the glycoproteins and glycolipids, we must have some fructose or mannose groups. So the mannose binding lectin, binds to it. It is a serum protein, it binds to it, and finally it causes the pathogenic destruction. Okay. In alternate pathway, a protein which is circulatory in function, it is called factor 1. Factor 1 binds to polysaccharides of cell membrane, and due to this binding, the three things which are activated, like okay, C3 and C5, two things, they are activated, the complement protein 3 and 5, which attack the pathogen and finally cause pathogenic destruction. So this is the whole antibody action. And now let's say discuss about the natural killer cells. You must have known natural killer cells. The function is virus destruction and kill the damaged cells, malignant cells. Okay, NK cells secrete cytokines too. Like many cytokines are secreted by natural killer cells, like IL, interleukin 2, and the INFs. Okay, then CSF, GM, CSF secrete the GM, CSF, and human necrosis factor alpha, TNF alpha. Okay, they are non-phagocytic. They have some enzymes too. They're hydrolytic enzymes like perforins. Perforins often cause a pore and pore or the holes in the cell membrane of the pathogen, and due to this pores occurs that the water enters into it. Water enters into it and it gets swelled up, which is called oncosis. Okay, oncosis is a process which occurs due to porphyrin action. And another enzyme which is present are the granzymes. Okay, these both are lytic enzymes. Okay, now let's talk about the cytokines. The cytokines are cell signaling molecules. These are the cell signaling molecules. Many types like platelet activating factors, interleukins, interferons, TNFs, cytokines, defensins, catalysogens. Okay, so in cytokines, the first one is platelet activating factor, PAF, which are secreted by the neutrophils and monocytes and cause the aggregation of platelets. Okay, the platelet aggregation occurs by this factor. Second is the interleukins. Interleukins are secreted by a variety of cells like T cells, B cells, eosinophils, basophils, mast cells, natural killer cells. All they secrete interleukins. Okay, interleukins may be are of 16 types, from IL-1 to IL-6, IL-16. Okay. Then interferons, we have interferons too, which are also called as INFs. Okay, these are glycoproteins and are of 3 types like IFN or INF. You can write INF2 and IFN2, both are acceptable. INF alpha or IFN alpha, IFN beta, and IFN gamma. Okay. Then we have some molecules called as chemokines, which are chemoattractants, and other one are defensins. We have alpha defensins and beta defensins. Okay, antimicrobial peptides are there. You must have remembered defensins, catalycidins, or antimicrobial. Catalycidins, these are also antimicrobial peptides. So these are the cytokines which are present. Okay, you may write that alpha defensins are present on neutrophils, Paneth cells secreted by. Beta defensins are secreted by the cutaneous cells, or the epithelial cells sometimes secrete the beta defensins. Okay, I'm not writing that. Now let's talk about an important topic which is called as immunization. Immunization may be of passive type or may be of active type. Okay, it may be of passive natural type or passive artificial type. Passive natural type, like the transfer of IgG from mother to the fetus. You must have remembered that IgG is the only antibody which can cross the placenta. This is the placental membrane. It can cross the placenta. Okay, so this is an example of passive natural type of immunization. Another is the colostrum. I'm writing only placenta, you must remember. One is the colostrum, the IgA. And the passive artificial type are the prophylactic measures. Okay, the prophylactic measures which are used for treatment against the tetanus, measles, diphtheria. Okay, now we'll talk about the active natural type of antibody immunization. So active natural type, like immune system produces antibodies which form the active natural type, and active artificial type of immunization. Okay, active natural and active artificial. In active artificial, again, 2 types are there. One is the vaccines, one is the toxoids. Okay, vaccines are maybe formed from, they may be live attenuated vaccines, they may be from dead pathogens. Okay, and toxoids, they elicit an immune response. These are the fragments of pathogens. Okay, pathogenic fragments. The first live vaccine was developed by Edward Jenner. There's a story behind it, the smallpox and the cowpox, and the complementation between the smallpox and the cowpox. So I discovered it, the first live batch. Lastly, we come to the immune deficiency diseases. Immune deficiency diseases. These are of two types, like congenital type or acquired. Congenital type may be of two types, like Digeorge syndrome. Okay, in Digeorge syndrome, actually it is the congenital absence of the thymus. Okay, congenital absence of thymus is called as Digeorge syndrome, absence of thymus. And second type is the SCID, severe combined immunodeficiency. This is the absence of lymphoid tissues. Okay, lymphoid tissues are absent, absence of lymphoid tissues. And in the acquired type, the most common is the AIDS, which is called as caused by the HIV virus. Okay, AIDS positive, we call the patient as positive case when the CD4 plus T cell count is less than 200 cells per mmq. The patient is called as the AIDS positive patient. So we have a lag period in AIDS. Okay, so these are that. About the safety of AIDS, we know awareness, blood screening is done, and we shouldn't share the needles, and we can prescribe a drug called as the Zidovudin. Okay, Zidovudin. So this is a drug of choice in the AIDS patients. The mode of action is by blood transfusion, is by sexual route or instruments or contaminated needles. Okay, the symptoms are fatigue, weight loss, chronic diarrhea, and ulcers. And HIV works by the process of reverse transcription. You must have known that HIV contains, the virus contains reverse transcriptase. Okay, reverse transcriptase. This was a phenomenon actually discovered by Temin in Baltimore. So it is called as Teminism, or the reverse central dogma of life. Central dogma, DNA, RNA, protein. So it's the reverse central dogma. And some opportunistic infections too are caused in AIDS, like pneumocystis carinii, and another is Kaposi sarcoma. Okay, other also there. Okay, not very important. So the HIV virus has glycoproteins, the spike proteins present over the outer coat, which interacts with the TCR, the monocyte receptors, the dendritic cell receptors. Okay, so this is all about the AIDS. And some other common autoimmune disorders are there. Okay, we haven't discussed yet about the autoimmune disorders. So let's discuss it. Autoimmune disorders. Autoimmune disorders. About the autoimmune disorders, these are caused by the self-antigens, in which the tolerance of the self-antigens often fails, or autoimmune disorders may be the abnormal antibodies are formed. Okay, this causes autoimmune disorders. They may be of organ-specific type or multi-systemic diseases. They may affect many systems, or they may be organ-specific. Okay. Some common are there like IDDM, insulin-dependent diabetes mellitus, in which the islet cell autoantibody is formed against the beta cells of the islets of Langerhans of the pancreas. Okay, and it may be caused by antibody against insulin and glutamic acid decarboxylase enzyme. Okay, or it may be due to activation of T cells against the islets. Many causes are there. Next is the myasthenia gravis. Myasthenia gravis. This is a neuromuscular disease, and autoantibody is generated against the ACH receptors. So, and again, another is Hashimoto thyroiditis. in late middle age, there's a commonly seen. And thyroid follicles here, thyroid follicles, they don't show activity. So activity of thyroid follicles cease in that disease. So we can see some hypothyroidism. Actually, antibodies are formed against the thyroid follicles, so self antibodies. Then another is the Graves' disease. Many names are there of Graves' disease, like we can call it the exophthalmic goiter and the Basedow disease or the Paris disease. Okay, this is the hyperthyroidism. And next is the rheumatoid arthritis. Rheumatoid arthritis. This is the chronic inflammation of synovial joints. Synovitis may occur. Okay, synovitis will occur. So the joints will be painful and stiff. So RA factors are produced. RA factors are the rheumatoid arthritis factors. So autoantibodies are produced against the rheumatoid. The autoantibodies are called as RA factors. They are produced against the synovial lining. Okay. Okay. In this video, we'll discuss about the allergy too. Okay. Allergy is a part of this topic only. So we are talking about allergy. Allergy is mainly the hypersensitivity. Okay. It is the hypersensitivity to certain allergens. Hypersensitivity. Which leads to immunological reactions or the immunological hypersensitivity reaction. Allergens are the agents that cause allergy, like they may be chemicals, they may be some food items, they may be pollen, or dust allergies may occur, drugs. Okay. There may be a variety of allergens. So hypersensitivity reactions or hypersensitive reactions are classified into 5 types. So type 1 is the first one, is the type 1 anaphylactic reactions. Okay. Second one is the type 2, which are the cytotoxic reactions. Third type is the antibody mediated reactions. Then the type 4 is cell mediated reactions. Fifth one is the type 5 is the stimulatory or blocking reactions. Okay. So let's discuss about the first type of reaction, that is let's say we are exposed to an allergen. Okay, I'm discussing the type 1. We are exposed to some allergen, so IgE is produced. IgE are also called, in the context of allergy, these are also called as reagents, or these are also called as sensitizing antibodies. Okay, they get bind to the mast cells. The IgE molecules get bind to the mast cells and macrophages, and provide the protection from allergy. Okay, cause allergy, cause the reaction. Type 2. In type 2, the cytotoxic reactions are majorly seen in the diseases like erythroblastosis fetalis, which is the RH disorder. You must know that the HDN is caused, and icterus and all, kernicterus. Or it can be seen in autoimmune hemolytic anemia. Okay, autoimmune hemolytic anemia, also seen in this. Okay, and these reactions involve IgG antibodies. Okay, So IgG antibodies bind to the blood cells and the affected cells are destroyed in that. Okay. So allergen and IgG reaction occurs and the affected cells are destroyed. Here IgE, the exact mode of action is that there are two cells here, and between between the receptors, the allergen is trapped. You may call it like the subsequent exposure. The first exposure is the second exposure. So in subsequent exposures, in the next exposure, the mast cells and the basophils are degranulated, and due to this degranulation, histamine is released, and histamine affects by increasing the BP, causes bronchoconstriction and shock. The state of shock is also caused by histamine. So these are the manifestations of the type 1 anaphylactic reaction. In type 3 reaction, let's discuss about the type 3 reaction too. In type 3 reaction, the antibody mediated reaction, there is an increase in production of Ig and increase in production of IgM, which are deposited in joints, cause arthritis. They're deposited in heart to cause myocarditis, and also deposited in glomeruli to form glomerulonephritis. Must have heard in the renal physiology we have glomerulonephritis, we have pyelonephritis, glomerulonephritis, also Bright's disease and all. Okay, and type 4 cell mediated immunity, or the reaction, cell mediated reaction. This is a form delayed reaction. It is delayed in nature, and in this the T lymphocytes are involved. Okay. So this is the delayed hypersensitivity, and this involves T lymphocytes. As it is a revision video, so we are not going to deep up the reactions and the examples associated. You can read it from the book. And the type 5 immunity, the reaction that is seen in autoimmune disorders. Disorders like Graves' disease or myasthenia gravis. Okay, like in myasthenia gravis, the TSH and the surface receptors of thyroid cells, the TSH stimulates it, and the surface receptor of thyroid cells, they cause the increase in the production of thyroid hormone. The B lymphocytes secrete TSA-B, that is thyroid stimulating antibody, which act by a positive feedback loop to increase the amount of thyroid hormone, and it causes Graves' disease. Okay, and in myasthenia gravis, IgG autoantibodies are developed. You know the procedure against the ACH and all. Okay, so allergy is like this only. And this covers all the essential concepts of allergy. Now we are going to discuss about the cancers. Cancer is the uncontrolled cellular proliferation in which the cells, they proliferate and not in a definite fashion, and malignant cells are proliferated. These cells are called as malignant cells, or neoplasm, you can say. And the pile of cells is called as the tumor. Tumor or neoplasm are synonymously used terms. Okay, so cancer of immune system, the cancer of immune system is called as leukemia or lymphoma, and the cancerous cells, they overpower the immune system. Okay, the immune system is rendered helpless in that, and the treatment includes like chemotherapy or radiotherapy, This chemotherapy and radiotherapy has effects on immune system too, because they weaken the immune system. Okay, immune system is weakened by chemotherapy and radiotherapy. And another is immunotherapy. And last is bone marrow transplant. Okay, so this is about a basic, just few lines about cancer and all. So let's discuss about platelets. We'll discuss about the platelets. We will discuss about the principles and the three sequence in which the hemostasis occurs and all. And lastly about the blood coagulation and the disorders, the bleeding disorders. Then we'll go to our blood transfusion and the blood grouping, and lastly jaundice. Okay, so the chapter will be completed in that. Platelets, they are called as thrombocytes. We know properties, like their size is about 2.5 mu and volume is 7.5 mu cube. They are small, colorless, non-nucleated. They are moderately refractile. Okay, there are many shapes, like the shapes may be dumbbell shape. Okay, it may be of dumbbell, it may be of comma shaped, or maybe of cigar shaped. Okay, irregularly shaped, rod shaped, or spherical. They may be. Thrombocyte in inactivated state don't have filopodia. Okay, filopodia is just the extensions. Okay, of the platelets. So in activated state, the filopodia are seen. Let's say this is the platelet. So if this is inactivated, and if it is activated, so they develop like that. These extensions are called as filopodia. Like you must have heard of the pseudopodia at amoeba and all. Filopodia. And filopodia, the function of filopodia is that they interlock between the platelets and help in its aggregation. Okay, help in aggregation of platelets. Now let's talk about the structure of the platelet. I'm drawing a simple structure. Okay. These are the canalicular system. Let's say this is the platelet. So we have some granules. We have some glycogen granules. We have microtubule. We have few granules called as the dense granules. Another granules are present, the alpha granules, and here we have mitochondria too in the platelets for energy production. So cell membrane is made up of phospholipids, cholesterol, glycolipids, carbohydrates as per the regular cell membrane and all. Glycoproteins help in adherence of platelets to collagen and endothelium. Phospholipids, they increase the clotting reaction. Okay, phospholipids of the platelets, phospholipids, they are the precursors of thromboxanes, mainly the thromboxane A2, prostaglandins, and some other substances. Okay, then we have the microtubules, which maintain the platelet shape, and the cytoplasm has a variety of components. We have to know the variety of components that are present in the cytoplasm. So let's say cytoplasm of platelets, they have a variety of compounds like proteins, they have enzymes, they have hormones, some granules. Okay, so let's see what are the substances that are present in the cytoplasm of the platelets. About the proteins, proteins. So proteins, we have some contractile proteins like actin, myosin, and thrombostatin. Thrombostatin is the contractile protein that helps in the clot retraction. Okay, we'll learn about them in the clotting part. Then we have von Willebrand factor. Von Willebrand factor. Von Willebrand factor actually increases the adhesiveness of platelets. Okay, the von Willebrand factor. Look, let's say this is the platelet. Damaged endothelium, the endothelium membrane which is damaged produces the von Willebrand factor, which interacts with the glycoprotein 1b in the platelets to increase the adhesiveness of the platelets. Okay, we'll see that in a few minutes. Then we have fibrin stabilizing factor. Fibrin stabilizing factor, that is the factor number 13. Okay. Then we have platelet derived growth factors which help in wound healing and blood vessel repair. Then we have platelet adhering factor which help in adhesion of platelets. And we have vitronectin. Vitronectin is also used in adhesion of platelets, and tissue cells are spread in culture due to vitronectin. Then we have thrombospondin. Thrombospondin, which prevents angiogenesis. Thrombospondin is the angiogenesis prevention. Okay, we have enzymes, a variety of enzymes like ATPases. We have ATPases. We have enzymes for synthesis of prostaglandins. Okay, then write a host of enzymes and all. Hormones, in hormones we have adrenaline and 5-hydroxytryptamine. You must have known the chemical name of serotonin. Then histamine. We have granules like the alpha granules and the dense granules. Okay, so we are talking about the dense granules and the alpha granules. Dense granules. The alpha granules consist of the components of the alpha granules include PDGF, VEGF, okay, and other compounds like endostatins and thrombospondin. And dense granules consist of nucleotides, the 5-hydroxytryptamine or serotonin, the phospholipids, calcium, and lysosomes and accumulation of that. Okay. The normal range of platelets is 2 to 4 lakh per mm cube, or 1.5 to 4 lakh per mm cube. We can have variations also in the platelet count. Normal count is 1.5 to 4 lakhs per mm cube. It is the normal. We can have variations we can have thrombocytopenia. It may be caused due to infections, leukemia, anemias and all. Then we have thrombocytosis in case of allergy, fever and trauma, and thrombocythemia. Thrombocythemia may be caused in carcinomas, in chronic leukemias, and in Hodgkin's disease. A disease is that Glanzmann thrombasthenia. Actually, the count is normal, but the clot retraction is deficit. Okay, the clot retraction is defective. Clot retraction. and due to this thrombocytopenia, purpura is occur. Okay, thrombocytopenic purpura occurs. About the critical count of platelets, the critical count is 10,000 mmQ. Okay, this is the critical count. Below critical count, we can have severe conditions. Then about the physiological variations in the normal count of platelets, physiological variations. This may depend on the age, like it is less in infants. It maximally reaches 2 lakhs and all. According to the gender, it decreases during the menstruation. Increases in high altitude and after meals the postprandial increase. Okay, now we have some properties of platelets. About the properties of platelets, we have 3 A's. Okay, we have 3 A's in the properties of platelets: adhesiveness, aggregation, and agglutination. Adhesiveness of platelets, we saw filopodia helps in it, and the collagen, thrombin, ADP, thromboxanes A2, cations, some proteins like P-selectins and vitronectin. This increase the adhesiveness of platelet. We saw that in Resolve increase the adhesiveness of platelets, and the platelets aggregate, and the aggregation of platelets are mediated or helped by filopodia. So filopodia help in the aggregation of platelets, and agglutination is the clumping. So they get clumped each other and form the clot. Okay, we'll see more about it in the hemostasis and coagulation mechanism. So blood loss prevention is done by hemostasis, and PDGF is useful for endothelial repair. Platelets have defensive mechanisms. Platelet help in blood clotting. They help in clot retraction and all. So platelet moderators, some are the moderators of platelet activity, some are activators, and some are the inhibitors. Okay, activators and inhibitors. The activators of platelet are generally P-selectins, the calcium, collagen, thrombin, the ADP, convulsin. Convulsin is actually the snake venom, the protein from snake venom. Okay, protein from snake venom. Some are the inhibitors of platelets, platelet activity, that is nitric oxide. Then we have clotting factors 2, 9, 10, 11, 12. So we have 9, 10, 11, 12 and 2. This help in this inhibit the platelets. Okay. Then we have prostacyclins and ADP breaking nucleotides, which help in inhibiting the platelets. Okay. So about the lifespan and fate of the platelets, first we can talk about the development. So it develops in the bone marrow. In development, it develops in the bone marrow. So pluripotent stem cells. Development of the platelets. Okay, the platelets derived from the bone marrow, and in the bone marrow pluripotent stem cells are present. So pluripotent stem cell, PPSCs. Okay, this give rise to CFUM. We have already saw it. Okay, and this in turn give rise to megakaryocytes. These megakaryocytes actually have pseudopodia. Okay, this pseudopodia are formed, and these are pinched off from their This get pinched off here and form some fragments. These fragments are called as platelets. Okay, these are called as platelets. So about the lifespan, the platelet have an average lifespan of 10 days. We can write the range as 8 to 11 days, and these are destroyed in the spleen. If there is splenomegaly, the increase in the size of spleen, the platelet count will decrease, and if the spleen is removed from the body as a splenectomy, the platelet count will increase. Okay. So now we are moving to the one of the most important topic, and the topic from which questions are generally expected, the hemostasis and the blood coagulation. So we are starting hemostasis. Actually, there are 3 steps in hemostasis. First step is called as vasoconstriction. The second step is known as platelet plug formation. And the third step is called as coagulation. So hemostasis is the spontaneous arrest of bleeding. So first of all, vasoconstriction occurs. That means the contraction of the small arteries or arterioles, they contract. Okay, and platelet are attached by von Willebrand factor. Then platelet plug formation occurs. In platelet plug formation, the platelet get adhered. Okay, the adhered platelets secrete, like this is the site of bleeding or injury, you can say, and these are the platelets that are adhered, and the adhered platelets secrete ADP and thromboxanes, and which in turn activate more platelets, and they attach here. Then again they secrete ADP, again more platelets are stimulated and they touch here. So you can say that it is a positive feedback loop. Okay, positive feedback loop. So due to this, if loose plug is formed, which is called as the hemostatic plug. So hemostatic plug formation occurs here, which closes the blood vessels, the narrow blood vessels or the small arteries, and the blood flow stops. Okay. Then we have the coagulation of blood, in which the fibrinogen is converted to fibrin, which forms threads, and they block the blood flow and all. Okay. So vasoconstriction is also mediated by serotonin. Serotonin is a vasoconstrictor, we know that, and aggregation of platelets occur, and prothrombin activator is activated. Blood clotting finally occurs. So finally, we are here in the blood coagulation topic. So the clot, what is the clot? Clot is a jelly-like material. Okay, it is a scab, or you can say some jelly-like substances which stop the blood flow. So blood loses its fluidity to form the jelly-like substance called as the clot. There are 13 factors involved in clotting. Okay, there are 13 factors. You must remember all the factors. So here we go. Let me make a trick. Okay, freshers party tonight, come, let's call seniors and call and some partying guys. Okay, so these are the factors. I am writing which one is the which. F stands for the fibrinogen, which is the clotting factor 1. P stands for prothrombin, which This is the clotting factor 2. T stands for thromboplastin. Thromboplastin. This is the clotting factor 3. C stands for calcium, which is the clotting factor 4. 5 stands for labile factor, or you can say that proacelerin. Okay, and this is the factor 5. About the factor 6, its presence is not proved. Okay, presence not proved. Although it's discovered, this is a story where when the sixth factor was its presence was not proved, could not be proved, although it was discovered. Okay, then S stands for stable factor or proconvertin. This is the clotting factor number 7. Then A stands for anti hemophilic factor A, AHFA, or anti-hemophilic globulin, you can say. This is the clotting factor number 8. C stands for Christmas factor, or AHFB, you can say, or it is called as PTC2. Okay, this is 9. S stands here again for Stewart Brower factor. This is the 10th. And this P, this guys is also only to make it suitable. Okay, but P stands for plasma thromboplastin antecedent plasma thromboplastin antecedent, or we can say it AHFC. To continue the trick, please freshers party tonight. Come, let's call seniors and call some partying guys. Okay, have fun. We have 2 more factors making the trick as have fun. H is Hegeman factor or contact factor or glass factor. Okay, glass factor. And F stands for fibrin stabilizing factor or fibrinase or Lackey Laurent factor. Okay, these are all the blood coagulation in which the factors are involved. Okay, we have 13 factors in total, or 12 factors, as 6th is not proved. And this is the trick. You can remember this by the trick. And then we have the enzyme cascade theory. It is one of the most important topic here. So the steps are to first formation of prothrombin activator. Okay, then prothrombin to thrombin and fibrinogen to fibrin. Okay, so prothrombin activator may be formed by the first step is formation of prothrombin activator, PTA, prothrombin activator. 2 pathways are there. One is intrinsic pathway and other is extrinsic pathway. Intrinsic pathway means the injury begins with blood. Okay, it begins in blood itself. And the extrinsic pathway begins with the tissue trauma. If there is a trauma in tissue, then extrinsic pathway is activated. Intrinsic pathway is obviously slower, and extrinsic pathway explosive. In 15 seconds it starts. Okay, so let's say we are taking the intrinsic pathway first. So let's say the blood vessel is injured, and first we are taking intrinsic one. So let's say there is an injury and blood vessels are ruptured and collagen is exposed. So collagen is there. This collagen stimulates the XII or the Hegeman factor. Okay, to convert into XIIA. This is the activated form. When I write some factor like XI and A means activated. Remember that. So this occurs in the presence of kallikrein, HMW, kinogen. Okay, This is the factor required for the conversion of XII to XIIA, which in turns activate XI to XIA, and this occurs in presence of HMW kinogen. And XIA then converts IX to IXA in presence of calcium, and this converts IXA, which converts X to XA in presence of factor 8 and calcium. Okay, so you have the activated XA. Now collagen is exposed, the platelet is activated. We have already looked into the activated platelet. It has philopodia. Okay, so the platelet, they synthesize, the activated platelet mainly synthesizes phospholipids. The phospholipids and the XIA, the XA, okay, this phospholipids and the XA here get combined in presence of calcium ion to form the prothrombin activator. Okay, from the prothrombin prothrombin activator. Let's look at the extrinsic pathway too. This is the intrinsic one, and let's look at the extrinsic one too. Extrinsic one. So the tissue is damaged. Tissue releases tissue thromboplastin. Tissue thromboplastin. You must have remembered that is the factor 3. Okay, and it contains glycoprotein and phospholipids, which convert the X into XA in presence of factor 7, and this XA reacts with 5 and calcium. It reacts with factor 5 and factor calcium, and finally these 3 form the prothrombin activator. So in either of the route we have the prothrombin activator. Okay, in both extrinsic as well as intrinsic pathway. So we have the first step done, formation of prothrombin activator. Then comes the second step. In the second step, the prothrombin activator converts prothrombin in presence of calcium to thrombin, which activates factor 5 to 5A. Okay, and this 5A again gives positive feedback to the prothrombin activator. So there exists a positive feedback loop here too. Positive feedback loop exists here too. Okay, then we'll see the formation of the conversion of fibrinogen to fibrin. The last step, fibrinogen to fibrin conversion. Okay, so the thrombin here, we have seen the formation of thrombin. This thrombin will stimulate the inactive fibrinogen to active fibrinogen, and this active fibrinogen polymerizes to form fibrin strands, and this fibrin strands in the presence of factor 13 and calcium form the clot. Okay, if the clot is formed externally, it is called a scab, and the clot has the composition of clot is RBC, WBC, platelets, fibrin framework is there. Okay, and the clot gets retracted within 30 to 45 minutes is the time for the clot retraction, and clot retraction is done by the thrombostatin. Okay, we saw in platelets the thrombostatin is present, contractile protein. Okay, so the clot dissolution is called as fibrinolysis. lysis. This is the clot dissolution. And this is done by plasmin. So let's see the mechanism through which it occurs. There is the liver. Liver secretes plasminogen. Plasminogen in presence of TPA and lysosomal enzymes form plasmin, and this plasmin forms plasmin. Okay, and this plasmin is responsible for the clot dissolution. Okay, so we have the anti-clotting mechanism too, because the endothelium and damaged tissue form the thrombomodulin, and the thrombomodulin thrombin complex is formed, which activate the protein C. The protein C in turn activates the gets activated. Okay, inactive protein C converts into active protein C, and in presence of protein S cofactor, it forms the factor 5, 8, and TPA inhibitor. Okay, and the TPA, the active TPA, forms plasminogen to plasmin. So this is the whole mechanism, the anti-clotting mechanism. Okay, this is not much needed. Okay, we can discuss it. Was anti-clotting mechanism. Anti-clotting mechanism. The anti-clotting mechanism, first of all, the endothelium and the damaged tissue. This from thrombomodulin. Like, let's say this is thrombomodulin, and this thrombomodulin reacts with thrombin. Let's say this is the thrombin. Okay, this gets fit in here. This forms the thrombomodulin thrombin complex, which activates protein C, and the protein C is activated into protein. This is the inactive form, and protein CA is the active form of protein C, and the protein C in presence of protein S cofactor, activated protein C in presence of protein S cofactor, which changes into TPA inhibitor. Okay, and the active TPA, the TPA gets activated. Okay, TPA inhibitor, so TPA gets activated, and it converts the plasminogen to plasmin. Okay, so this is the anti-clotting mechanism. Some natural anticoagulants are there in blood. Some natural anticoagulants. We have heard the example of heparin. Others are there like protein C. Third is antithrombin 3, and fourth is alpha 2 macroglobulin. Okay. We'll look at some of the anticoagulants. Okay, the first one is heparin. Heparin is produced by the mast cells. It is a conjugated polysaccharide. It works in vivo as well as in vitro. Actually, what it does, it suppresses the thrombin. So clotting will not occur. Then it can combine with antithrombin 3 factor, antithrombin 3. It may combine with that and activate it to stop the clot, or it may inactivate 9, 10, 11 and 12 clotting factors. Okay, so these are the functions. Clinical uses in intravascular clotting and surgery, in dialysis, or in preserving blood before transfusion and all. Laboratory use, so we use 0.2 to 0.8 milligram for 1 ml blood. Okay, this is the laboratory uses of heparin. And heparin is one of the most expensive anticoagulant. Okay, so we have another derivatives which are called as coumarin derivatives. In coumarin derivatives, we have two types like warfarin. Warfarin is also used to prevent the myocardial infarctions or strokes and thrombosis all. And the second one is dicumarol. And dicumarol is the oral anticoagulant. Okay, remember it, oral anticoagulant. It inactivates mainly the vitamin K, which is essential for the formation of clotting factors 2, 7, 9 and 10. This is not important much. Okay, I can write it. Okay, vitamin K is essential for formation of clotting factors 2, 7, 9 and 10. Okay, this is an important statement, please remember it. Second is EDTA. The third one is EDTA, ethylene diamine tetraacetic acid. Okay, so let's see its usage. EDTA. Okay, we can have like Na2 EDTA complex, or we can have K3 EDTA. Okay, EDTA, what it does, it removes the calcium by forming a chelate ring. You must have seen the EDTA structure in chemistry in the previous classes. Okay, so it acts as an anticoagulant in the laboratory, and intravenous EDTA is injected in form of lead poisoning. In case of PV poisoning, IV of EDTA. EDTA is given as IV. Okay, intravenously. Then we have oxalate compounds too. We have some oxalates like calcium oxalate. Calcium oxalate is actually the PPT, which is formed due to oxalate compounds. Okay, this reduces the blood calcium level, which in turn can help in the anti-clotting. Then second is citrates like sodium citrate or ammonium citrate or K plus citrates. This form insoluble calcium citrates. These are also used in vitro, in blood banks. In blood banks, what we do, we use acid citrate dextrose, which are mixed in the ratio 1:4, and CPD2 like citrate phosphate dextrose, which are mixed with blood in the same ratio opposite to that, like 4:1. These are all. In laboratory, we use a solution which is called as the DACIS solution, DACIS solution. DACIS solution is used majorly to determine the RBC platelet count. So here the formal citrate solution is used. Okay. Some other things are there which prevent blood clotting, like peptones and C-type lectins. C-type lectins are mainly found in the venom of viper. Okay, the viper is a snake, and the venom of viper contains the C-type lectins. And hirudin is from hirudinaria, the leech. Okay, and some physical methods are there, like cold treatment. We can use cold treatment, and the blood is collected in the silicon coated container, because factor 12 is absent. Okay, we collect blood silicon coated container. Why so? Because it absence of factor 12. Okay, now we have some procoagulants or hemostatic agents which facilitate the hemostasis. Okay, like we have thrombin, increase the blood clotting, and snake venom, like proteolytic enzymes from snake venom. You can write simply snake venom. You can write lungs and thymus extracts. Okay, tissue extracts majorly, because tissue have thromboplastin, which increase the blood clotting. This also increase, this also increase. We have some sodium and calcium arginate, which activates the H1 factor. Okay, this increase the blood clotting. And oxidized cellulose. This all. So the work of them are to increase to these are called as hemostatic agents or procoagulants. We can determine the some tests are there for blood clotting, like we have the bleeding time test, bleeding time, and the method used is called as Duke's method. So the normal bleeding time is 3 to 6 minutes. This is the normal count, and we have some test called clotting time test, which is determined by capillary tube method, and normal is 3 to 8 minutes. There is a delay, you can see from bleeding time, clotting time. We have PT, prothrombin time, and the normal range is 10 to 12 seconds. Okay, the PT is when blood is added to the tissue thromboplastin to the formation of clot, is the prothrombin time. Okay, and bleeding time is the oozing starts, when the oozing of blood starts, to the arrest of bleeding. Okay, and we have a thing called as partial or activated thromboplastin time, activated prothrombin time, sorry, PPT, partial thromboplastin time, we can say that, or we can say activated prothrombin time. Okay, PPT or APPT or APT. Okay, this is normally 30 to 40 seconds. So this is the time duration in which blood, from oozing of blood to the clotting. But here the platelet substitute is phospholipids. Phospholipids and calcium are given. Okay. We have a thing called as international normalized ratio, INR, international normalized ratio. This is the rating of PT with respect to an average. Okay, we compare the PT, prothrombin time, with an average, you compare it. Okay, so it is normally 1, and if INR is greater than 4, there is uncontrolled clotting risk. Okay, clotting will not occur, uncontrolled clotting. We have thrombin time too. If blood is activated by thrombin till the formation of clot, we have the thrombin time, TT. So these are the some of the tests which are employed for the blood clotting. And now let's discuss into the clinical and clinical discussion. Just a minute. Okay, so we're discussing about the clinical correlations of blood clotting. Obviously these all are clinical, but we have few diseases and bleeding disorders, like we have a disease called thrombosis. Okay, you must have known about the thrombus and the emboli. Let's say this is the blood vessel, so a mass is formed due to intravascular blood clotting. This is called as thrombus, and the moving thrombus, if the thrombus gets mobilized, if it moves away through the blood, it travels by the blood, so it is called as emboli. Okay, so there is a difference between thrombus, and thrombus is the solid mass, whereas emboli is the moving mass. So the phenomenon is called as embolism. We may have pulmonary embolism, we may have cerebral embolism, or coronary embolism. Okay, depending upon the location to which it moves. There are many causes of thrombosis, like RBC agglutination. RBC may agglutinate due to any abnormality or all, due to endothelial destruction, like and this may be due to like bedridden patients, because bedridden patients have sluggish blood flow. Their blood flow is very slow, so they may have thrombosis. Or you can have protein C absence due to his the clotting factors 5 and 8 are not produced, so we can have that also. Okay. So complications, what are complications? That embolism is very dangerous. One of them, ischemia, due to which the blood vessel is obstructed and tissue is damaged. Necrosis, which causes the tissue death. Okay, so we call necrosis a tissue death. And infarction occurs. Infarction, like myocardial infarction, or infarction may occur in many organs like kidney, lung, brain, heart, spleen. We know the myocardial infarction, of course. And the infarct, okay, a term is there, infarct. Infarct is the specific region of the tissue that is damaged. Tissue that is damaged. So we have some drugs like to treat the thrombosis. So you can prescribe aspirin. Aspirin is banned now. Okay, dextrans. So these are the drugs to treat thromboembolism. Okay, we have some bleeding disorders. Let's discuss them. Bleeding disorders. Some bleeding disorders like hemophilia. First one, very important. Hemophilia. Hemophilia is the cause may be due to there are many types of hemophilia like hemophilia A, hemophilia B, or hemophilia C. Hemophilia A is due to clotting factor 8 deficiency. Hemophilia B is due to clotting factor 9th deficiency, and C is due to the 11th deficiency. Okay. Next is hemophilia C is the rarest. Hemophilia A is also called as the classic hemophilia. Hemophilia is known by various names, like it's called as the bleeder's disease, or some may call it that the royal disease, because it is found in the Victorian ancestry and all. So like that. Hemophilia B is the Christmas disease, because the 9th factor we saw above. Here we have the 9th factor, which is called as the Christmas factor. So the hemophilia B is called as the Christmas disease. Christmas disease. Okay. And hemophilia A is the most common, and hemophilia C is the least common, the rarest. Symptoms: there is spontaneous bleeding, and the bleeding is prolonged. There may occur hemorrhage. Okay. Hemorrhages. And the joints may bleed, the painful joints, and blood in urine. Joints bleed. These are some of the symptoms of this hemophilia, and the treatment includes the replacing the missing clotting factor, as there is no other way. We can replace the clotting factor, obviously. Then we have another disorder called as the purpura. I'm sure you have heard about it. Purpura, in which the bleeding time BT is prolonged. The bleeding is prolonged, but the clotting time remains normal. Okay. Here the spots are seen, like hemorrhagic spots are seen, which are called as purpuric spots. These are the characteristic feature of this purpura, like purplish spots which are seen under the skin. Okay. So this is the thing. If blood is collected in large areas within the skin, if not in purpura but in other disorders in which blood is collected within the skin in very large areas, these are called as ecchymosis. Okay. Ecchymosis are large collections of blood within the skin, and we're discussing on purpura, obviously. So purpura, there are three types majorly. Like one is the thrombocytopenic purpura due to platelet deficit. Okay, this is due to platelet deficit. Second one is idiopathic thrombocytopenic purpura, where there is the term idiopathic, means the cause is unknown. Okay. Then we have the thrombasthenic purpura. This is due to the structural and functional abnormality of the platelets. Okay. Then we have a third disorder called as the Van Willebrand disease. The Von Willebrand disease, and you must have remembered the Van Willebrand factor, which helps in the adherence of the platelets. So there will be decreased platelet adhesion, and this will lead to excessive bleeding. Okay, this is the thing, Von Willebrand disease. So with that, we discussed about the bleeding disorders, the clotting disorders. Okay, so that's it. In the next video, we'll discuss about the blood grouping and blood transfusion. Okay, there's a separate branch called the transfusion medicine and all. So we'll not discuss about the medicine, just about the blood transfusion, the basic idea. And lastly, we'll cover the jaundice part, because jaundice is often covered in the GIT physiology. Okay, but we'll cover it here as Guyton has given it. So after jaundice, the chapter will be completed. Okay, so that's all. Thank you. This video, it is a continuation of the hematology. So in this video, we are going to discuss about the blood grouping, okay, and the transfusion, and lastly we'll go to the jaundice part. So today the chapter will be finished. Okay, the whole unit of hematology in detail. Let's start. So we have blood group, blood cell antigens. Okay, the blood grouping, the topic of blood grouping revolves around the antigens. Okay, the blood group antigens. So we have many antigens, like we have antigens on RBC, WBC, platelets, plasma proteins. Plasma proteins are protein antigens, while RBC, WBC, and platelets are polysaccharide antigens. Okay, like we have platelet antigens too. We have platelet antigens, just seen upon the platelet membrane. Human platelet antigen is called that, and it has mild to moderate immune response, and it is less antigenic. Okay, it's not life-threatening and all. Then we can have HLA, human leukocyte antigen. These are seen in all tissues and organs, seen in WBCs, are of 150 types. Okay, every human has only 6 antigens, which translates to nearly 144 antibodies. While HLA has high immune responsiveness, it is potent and virulent. That's why we use HLA. We use the matching called the HLA matching in organ transplantation. Okay, it is not needed for blood transfusion. For blood transfusion, it is not needed, but it is important, very important for organ transplantation, because rejection of organ may occur if HLA matching or tissue typing is not done. Okay. Secondly, we have RBC antigens. We have about 36 different systems and more than 300 antigens. We have the systems, the major systems, the ABO, the RH, the Lewis system, the Kell system, the Duffy. These are rare systems, the LMN, and the I system. This ABO and RH, these are the major ones, while this may be found in minor or rare cases. Okay. Antigens we know are agglutinogens, they are polysaccharides, while antibodies are isoagglutinins. They may be of natural antibody or acquired antibodies. Okay, antibodies may be of two types. They may be natural, which are IgM category, or they are acquired, which belong to IgG category. So as they are natural antibodies, we often call them the cold antibodies, and the IgG category, the acquired antibodies, are called as warm antibodies. When we're discussing about the blood grouping, a law is very famous. It is called as the Landsteiner's law. Landstein's law talks about the reciprocal relations of the antigens and antibodies present in the plasma. If the antigen present, let's say A antigen is present, so corresponding antibody is not found. Okay, so anti-A antibody is not present in plasma. If antigen is absent, let's say A antigen is not present, so anti-A antibody is present in plasma. And the third statement is that absence or presence of Rh antigens doesn't depend. No anti-Rh antibodies are present. No anti-Rh antibodies are present. No matter what, no anti-Rh antibodies are present. Okay, so Landsteiner was the father of the blood grouping. Okay, but Landsteiner law has the exception to, like Rh system has the exception of Landsteiner's law. Okay, it is the exceptional statement here, was added later on. Okay, so cold antibodies and warm antibodies, you must not have understood the concept. The cold antibodies are IgM type. They're called like that because they occur only at temperature less than 37 degrees Celsius. The agglutination, the action, occurs at temperature less than 37 degree temperature, while the agglutination occurs at only at 37 degree Celsius, or the room temperature, body temperature, in case of warm antibodies. Okay, so we will discuss the ABO system majorly because it is in our syllabus. The ABO system. In the ABO system, let's say this is the RBC membrane. This is the RBC membrane. Okay, so there are two units here. There are two units. These units are called as fucose. FUCOS, fucose. And here we have the A antigen, N-acetyl galactosamine, which is the A antigen, and here we have the galactose, which is the B antigen. Okay, so if somebody has A blood group, he may have the phenotype is A and the genotype may be IgA, not IgA, it may write as IA, it may be IAI or IAIO. The corresponding antibody is anti-B antibody. Okay, and if somebody has B blood group, the genotype may be IBIB or IBI, and the anti-A antibody are present in the plasma. In case of AB blood group, IA, IB. Okay, and here both antigens are present but no antibody. So this is the universal recipient. Okay, and O blood group, only fucose is present, but IO it is written, or you can simply write II. Okay, and this have both anti-A as well as anti-B present in that. Okay, so these are the basic principles of the blood groups. Then we have a blood group called as the Bombay blood group. Here the fucose is absent. And no NAG, N-acetyl glucosamine is also absent, and the Galactose is also absent. Okay, all are absent. No antigens on RBC membrane. Okay, so if the patient's test his blood, so there will be O in the result. No energy, no galactose. Okay, but antibodies are there. Antibodies will be anti-fucose antibody, anti-fucose, anti-NAG. Form blood group is rare, pretty rare. Okay, and anti-galactose antibodies are present. Okay, then we have the RH system. RH system is represented by the D antigen. This is the most common antigen, but we can have like C, D, E, and the analogs small c, small d, and small e. Okay, 6 common types are there, but D antigen is the most antigenic and widely found too. This is the most antigenic. So these are the okay. Discuss about the major systems, the ABO and the RH. The minor systems which cause mild transfusion reactions. Some minor systems we can add, like we have written major systems as ABO and RH, and minor as Lewis, Kell, Duffy, Ellerman, and I system. Okay, this Kell and Kelly and Duffy, these systems are also called as familial systems. This both are called as familial. Just found in few families. Okay, this is rare. And other systems are there, like MNS system. Okay, and we have P system and all. These are not in our syllabus, so not read them. Okay, the antigens appearance of antigens and antibodies. The antigens, blood group antigens, appear in 6th week of fetal life. Okay, and the antibodies, they appear 10 to 15 days after birth. Okay, the maximum concentration is reached at the 10 years. Okay, so this is about the appearance. About the RH grouping, we told that the D antigen is the most antigenic. Okay, it was first discovered, the RH stands for rhesus. Okay, so it was first discovered in the rhesus monkey, discovered by Landsteiner and Wiener. Okay, in 1940. CDE are the types, and 6 common types we said. D is the most commonest, most antigenic, and the antigens are the antibodies are the warm antibodies. Okay, because they work at the temperature of 37 degrees Celsius. And RH antigens are of D antigen is of IgG type. They can cross placenta, so this will cause some diseases too, like we must have heard of HTN, the hemolytic disease of newborn. Okay, so HTN has some manifestations. The manifestations are, there are some manifestations of HTN, like we can have manifestations like erythroblastosis fetalis, we can have kernicterus. In kernicterus, the basal ganglia of the brain, you must have known about the limbic system. The basal ganglia of the brain, the bilirubin is deposited. Okay, there's the bilirubin deposition in the basal ganglia. This is the bilirubin increase in basal ganglia. Okay, erythroblastosis fetalis, it is common in the second pregnancy, where the mother may be injected with the sort called as Rogam sort. Okay, the injection that is administered to prevent it is Rogam. And we may have another manifestation, which is icterus gravis, due to which jaundice occurs. And we can have hydrops fetalis too. Hydrops fetalis, due to which the fetus is grossly edematous. Okay, grossly edematous fetus. Grossly edematous fetus is formed. So clinical applications we saw like blood transfusion and HDL prevention, paternity disputes to solve, medical legal cases to. Okay. Then we are coming to the next topic after the blood grouping. We are discussing the blood transfusion. Okay. Blood transfusion, in which the blood is donated from the donor to the recipient. We know the universal recipient is AB. AB positive is the universal recipient, while the O negative is the universal donor. Okay. So indications of blood transfusion, the causes, the diseases in which blood transfusion is needed, like in peptic ulcers, because in peptic ulcers there is a chronic bleeding, the blood gets released at regular intervals and all, so we need blood transfusion. Like another disorder is aortic aneurysm. There occurs a ballooning in the aorta and all, so aortic aneurysm. In ectopic pregnancy too. In different diseases like leukemia, purpura, in the clotting defects, in aplastic anemia. Okay, these all are the indications of blood transfusion. So we should have the selection of a healthy donor. Okay, healthy donor. A healthy donor must be healthy in all respects. He or she may be of 18 to 60 years. Okay, should not be a pregnant or lactating mother, and must be screened for HIV, hepatitis, malaria, okay, syphilis. Screening must be done. The hemoglobin concentration in the PCV must be normal. Okay, these are the contraindications of a. These are the features of a healthy donor. These are precautions for selecting a donor. Okay. We should take the precautions during blood transfusion, like we should take the aseptic measures. We should carefully watch, and the blood bag must be checked before transfusion and all. Okay. So mismatched transfusion causes various symptoms, like mismatched transfusion may cause diseases like ischemia, hemoglobinemia, hemolytic jaundice, renal vasoconstriction, circulatory shock, or renal tubular damage. Okay. We can have the various hazards of blood transfusion, like hypervolemia. We can get bloodborne infections like AIDS and viral hepatitis. Okay. We can have allergies like rashes, and asthma may be caused. Okay. Reduced tissue oxygen may be caused due to low 2,3-diphosphoglyceric acid, and hemosiderosis may be caused due to Fe overload, iron overload in liver and heart. Okay. We can have thrombophlebitis at the venipuncture site where from where the blood is taken, or we can have air embolism. The air bubble may be formed and it may move through the bloodstream and all. So, autologous blood transfusion, the term is autologous blood transfusion. Autologous blood transfusion. In this procedure, the transfusion of individual's own blood is done. Okay, the own blood of individual is withdrawn, stored. Okay, example in surgeries and in sportsmen. This is commonly done in surgeries and in sportsmen. Autologous blood transfusion is done. It is the individual's own blood which was stored earlier. Okay. And storage of blood in transfusion. So we can store blood for 20 ml of blood in 1 unit is added with 120 ml of ACD solution. ACD solutions contains acid citrate, trisodium citrate, dextrose, distilled water, etc. Okay. And one can donate 1 unit of blood every 6 months. Okay, and the blood can be stored for at least like 21 days. The blood can be stored. WBC platelets are get absent after 24 hours of storage, and RBC generally 1% RBC is destructed per day, 1% RBC destroyed in a day. Okay, the rate of destruction of RBC when the blood is stored. Okay. Then we will move to the last topic, our last discussion in this unit, which is not essentially of this, but as bilirubin and biliverdin etc. are discussed, so we'll discuss this also. So we are discussing about the jaundice. Okay, it comes as a long question too. Okay, so in heme iron and biliverdin, we know that the biliverdin, in presence of biliverdin reductase enzyme, get converted into bilirubin. Okay, so jaundice is the yellowish discoloration of the skin, sclera and mucous membrane. Okay, it is the yellow coloration of skin, sclera of the eye, white of the eye, that becomes yellow, the mucous membrane. It may be caused due to increased bilirubin concentration or the hyperbilirubinemia. Okay, the normal range of bilirubin is the normal range is 0.2 to 0.8 milligram per 100 millimeter of blood, but in case of this jaundice, it increases to 2 milligram per 100 ml. This is the abnormal. Okay, if it increases from 1.5 milligram per dl, the skin starts yellowing. So we must remember the facts here. Okay, so in hemoglobin, as hemoglobin gets destroyed, the globin is formed. Globin goes to the amino acid pool. Iron is stored at ferritin and sometimes at hemosiderin and all. We saw that. And the biliverdin gets converted to bilirubin. Okay, and the bilirubin is taken by the liver. Say this is the biliverdin. This in the presence of bilirubin reductase and then here. Okay, gets converted to bilirubin. This is taken up by liver. Liver takes this. Albumin removed. Actually, bilirubin transports in blood in combination with albumin. Okay, so this is called as the conjugated bilirubin, conjugated form. So here albumin is removed, so it is the unconjugated form. Okay, that's a mistake for that. Here bilirubin combines with albumin, but this is not exactly the conjugated form. Okay, I wrote it by mistake here. Okay, the unconjugated form, this is the unconjugated form of bilirubin, so we can write it as Bu, we can write it unconjugated bilirubin. Okay, this gets conjugated, this combines with UDP, glucuronic acid. Okay, and in presence of UDP, glucuronic transferase, it converts into conjugated bilirubin, and this conjugated bilirubin is excreted from the bile canaliculi and enters the intestine by active transport and all. Okay, in intestine, as it reaches intestine, the conjugated bilirubin reaches the intestine, and in intestine the conjugated bilirubin in presence of beta glucuronidase gets converted to urobilinogen, urobilinogen and stercobilinogen. Urobilinogen is excreted in urine and stercobilinogen in feces, obviously. Okay. So we have the etiology of jaundice. Okay, the classification of the etiology. So jaundice may be classified of three types. First is the prehepatic or the hemolytic jaundice. The prehepatic or the hemolytic jaundice, it is also called as unconjugated jaundice, unconjugated hyperbilirubinemia, okay, because all the bilirubin are in unconjugated form. Okay, it's not yet conjugated. Okay, so mechanism is that the RBC breakdown increases, the cause you can say that before reaching the liver where the increase in the bilirubin before reaching the liver causes the prehepatic or the unconjugated hyperbilirubinemia. Okay, and we have a test called as the Vandenberg test. We will discuss that slight detail. So this is the prehepatic or hemolytic jaundice. Second is the hepatic or hepatocellular jaundice, hepatic or hepatocellular jaundice. Here the liver cells are destroyed, the damage to liver cells. And the third is the post-hepatic or the cholestatic jaundice. In this, the bile duct is obstructed, Okay, now we can discuss about the test because it is useful in the differentiating all the jaundices and all. This is called as the Vandenberg test. The reagent uses the diazo reagent. Diazo reagent contains of three parts: sulfonylnic acid, HCl, and sodium nitrate. This may be of two types. Vandenberg test may be direct test or it may be like indirect test. In direct test, the diazo reagent plus the serum gives red brown color, in 30 seconds, while in indirect test, no color, diazotizant and serum give no color. If alcohol is added, they give red brown color, and it is a bit delayed. Okay, brown, it's a bit delayed. So like Vandenberg test, we saw that about the Vandenberg test, and the specific blood tests to like in prehepatic or the unconjugated hyperbilirubinemia, the peripheral blood smear is the normal. Okay, the albumin globulin ratio is normal, the alkaline phosphatase is normal, and the liver function tests, these are also normal. Okay, in hepatocellular or the choleric jaundice, it is also called as choleric jaundice. Choleric. Okay. This is caused due to liver damage, the liver cirrhosis or hepatitis and all. So in this, both conjugated as well as unconjugated bilirubin increase in level. Okay, so we can write it increase in levels of both conjugated and unconjugated bilirubin. Okay, they increase in level. Then the fecal stercobilinogen is less, so pale yellow stools are passed in that. So in Vandenberg test, both biphagic reaction is seen. So biphagic reaction seen in Vandenberg test. Okay, I have written VDB test. Please understand it. Here's in the specific blood tests. In case of the hepatic jaundice, for the specific blood test, the peripheral blood smear is the normal, the liver function tests are impaired, that abnormal, and the albumin by globulin ratio is decreased, very important. Okay, now we have the post-hepatic or the cholestatic jaundice, also called as the obstructive jaundice. Obstructive jaundice. Okay, obstructive jaundice, this is called as the conjugated hyperbilirubinemia because all the bilirubin becomes the conjugated type. The cause is due to the bile duct obstruction. The Vandenberg test is usually direct test. Okay, color appears in 30 seconds. Urine urobilinogen may be decreased or absent. Here the alkaline phosphatase is positive. Okay, there is more alkaline phosphatase, and rest all like peripheral blood smear, albumin globulin ratio, liver function tests are normal. Okay, here the stools, about the characteristic color of stools, they are of clay colored stools. Okay, clay colored stools are passed. Or about the color, we some books write at echolic phases due to stercobilin, lack of stercobilin and other bile pigments. Okay, the fecal fat level also increases, and the total obstructive jaundice, in total obstructive jaundice, the test for urobilinogen is also completely negative. Okay, last topic of the last part is the neonatal jaundice. You might have seen the neonates being kept in the room. Okay, and prescribed with phenobarbital drugs and all. So phototherapy is given to them, right? White light after the birth. Actually, what happens after the birth? There is the increase in the RBC destruction. We saw in the very first discussion that about from 6 million per cubic mm, RBC reduces to the 5.5 and 5. So there is a rapid destruction in RBC. So this is also called as the physiological jaundice. and the drug of choice is phenobarbital. Okay, this increase the activity of gluconeorin transferase, that is of okay high concepts not. The neonate becomes yellow. The There is another alternative method we use commonly, that is phototherapy. In phototherapy, what happens is that the bilirubin, there's the bilirubin accumulation, so the bilirubin gets converted to lumirubin. Which is water soluble, and it gets excreted with bile. Okay, it gets excreted out with bile, so the neonates get healthier. And neonatal jaundice is commonly seen after 2 to 5 days after the birth. Okay, so cause is the same, the RBC destruction, and sometimes we give the cause of the hepatic immaturity. So that's all, guys. In the very long video, I have covered it in parts and recorded it in parts, but hats off to those who are continually watching this, and we have successfully completed the hematology. I believe that I have covered all the parts from Gaiter as well as Genome, and as per PZ references are concerned. Okay, some you can always refer the book and the FASTED for USMLE prep or PLAB prep. So this video is useful for the as per the PG prep is concerned or the NExT prep is concerned, and those who are for preparing for the Prof examinations, they have to watch the video, because you have to cover the concepts in the very small time. Okay, so that's all. Let's meet in yet another topic, maybe nerve muscle physio, or we will take up a lecture on embryology and all. That's all for now. Thank you.